going to start very soon rather than making some setup. So once setup is going to be over, then you will be able to this going to be starts. My request to you all, keep silence in the class. Because when the session starts, don't disturb any other people. Please listen that what points you can add to your presentation. And then they start by listening to that talk with listening to the other students. So that how you can improve myself in the better way of presentation. That you can just take it off. And please keep silence while going for the presentation.
This okay. 6 seconds.
starting our session now. So you have 8 minutes for your presentation and 2 minutes for question and answer from the judges. Okay? We'll have a bell. The first bell states that your 8 minutes is completed. The second bell is for the 10 minutes. Okay? After that, you have to stop your presentation. Try to finish it in 8 minutes. Okay? Done. Good morning. You all have high energy. Ready for your presentation? Okay. We'll start. First, all these three people will be commentators. I'll be giving you one mic. Three of you are presently just pass the mic and present it. Okay? Be comfortable in whatever the positions you want to be in, whichever side you want to go. Is that okay? All the very best.
Good morning everyone. Myself Ujjwala with my teammates Darshini and Chaitanya from Primary High School. We thank Government of India to give me this opportunity to present our project analyzing food miles of selected vegetables and fruits consumed in river colony. Food mile measures the distance food travels from its place of production to the consumer. This concept is crucial in assessing the environmental impact, including greenhouse gas emission, energy consumption, and overall sustainability. Understanding and minimizing food can contribute to environmental conservation and human well-being. To identify the sources of vegetables and fruits consumed in reverse colony, to calculate the distance traveled by each food item to reverse colony, to analyze the environmental impact of these food miles. Use geographical mapping tools to calculate the distance traveled by each food item to reverse colony. Compare and analyze the data to determine their journey from farms to market. Conducted 100 surveys and interviews with local residents and market vendors to gather information on the origin of selected vegetables and fruits. Tracking the supply chain of selected vegetables and fruits to determine their journey from farms to market. Fruits and vegetables travel to reverse colony. These are our average food mile calculation. Onion. Onion is from Bijapur, Chitradurga, Davangere. This is the calculation and the answer is 347.33 kilometers. Similarly, we calculated other vegetables and fruits.
Environmental impact assessment. Estimation of carbon footprint associated with the transportation of this food meals using standard emission factors. Assumptions. The average weight of transported vegetables and fruits is approximately 110. Emission factor 0.1 kg and carbon footprint per ton kilometer. And calculation. The carbon footprint CF can be calculated using the formula CF is equal to distance into weight into emission factor, given that the weight is 110 and the emission factor is 0.1 kg. Carbon dioxide per ton kilometer. The formula simplifies to CF is equals to distance into 0.1. Calculation of carbon footprint for each item. Onion. Average distance is 347.33 kilometer and the CF is equals to distance into weight in emission factor is equals to 34.333 kilometer carbon dioxide. Similarly, we calculated the other selected vegetables and fruits. Summary of carbon footprint. average weight of transported vegetables and fruits is 1 ton. Energy consumption factor for road transportation approximately 2.3 mJ megajoule per 10 kilometer. Calculation: the energy consumption EC can be calculated using the formula EC is equals to distance into weight into energy consumption factor. Given that the weight is 1 ton and the energy consumption factor is 2.3 megajoule per 10 kilometer, the formula simplifies to EC is equals to distance into 2.3. Energy consumption for each item: onion, average distance is 347.33 kilometers and the EC is equals to 347.33 into 2.3 is equals to 798.859 megajoule. Similarly, we calculated the other selected vegetables and fruits. This is the summary of energy consumption. And this is the average distance kilometers and this is the energy consumption megajoule and this is the bar graph. The low impact food items. Relatively lower transported distances make it lower impact. Source location: papaya, Hoskote, Anigal and Devanahalli, and average distance traveled 42.33 km and carbon footprint 4.233 kg of carbon dioxide and energy consumption 97.359 megajoule. Significance for human well-being: reducing food waste can lead to lower greenhouse gas emissions and energy consumption, contributing to a healthier environment. Supporting local farmers and markets can boost the local economy and provide fresher produce to consumers. Consuming locally grown vegetables and fruits often means fresher and more nutritious food options, leading to better health outcomes.
Food pellets analyze in vegetables. The analysis of food pellets for vegetables and fruits consumed in kilos highlights the significant role that food transportations places in contributing to climate change and its impact to human well-being. High impact food item which travel long distance to greenhouse gas emissions and energy consumption. These effect negatively impact both the environment and public health. Reducing food waste through local sourcing, supporting farmers market, and encouraging seasonal production, and can mitigate climate change. These strategies foster a more sustainable food system and enhance community well-being. Benefits include environmental conservation, economic advantage, improved human health from fresher, more nutritious food, and better overall quality of life for local production. We thank our HM and guide teachers for allowing to us conduct the study. We also thank Sri Mithila sir and Sri Chandrabhai sir for guiding us. Our gratitude extends to the scientists, researchers and experts of the energy wetlands research group entered for for Center for Ecology Science, India Institute of Science, the FDRMSS Bangalore for their support.
And this is the reference.
Your calculation, footprint of apple appears to be highest. What's the lesson you derive? What is going to convey? Why it is so, and what is the outcome of that? What's the conclusion?
Speak aloud.
You have given calculation for footprint of different fruits and vegetables. From the calculation, we concluded that the footprint of apple is the highest and compared to other fruits and vegetables. What's the lesson you derive? Bar chart. Yeah. So apple is the highest energy consumption. So what's the lesson you derive from it? What's the conclusion? What do you want to convey by this? What are you conveying?
And answering.
Hey, no issue. You can take your own language. Language, no problem.
Sir, apple ಅಷ್ಟು ದಿನ ಬರ್ತಿದೆ ನಾವು ಅದನ್ನ ಅದ್ರದ್ದೇ ನ್ಯೂಟ್ರಿಯೆಂಟ್ಗಳನ್ನ ಬೇರೆ ಫ್ರೂಟ್ ತಿನ್ಬೋದು. ಈ ಲೋಕಲ್ ಅಲ್ಲಿ ಯಾವ್ದು ಗ್ರೋನ್ ಆಗುತ್ತೋ ಆ ಫ್ರೂಟ್ ನ ತಿನ್ಬೋದು. ಮತ್ತೆ ಅದರಿಂದ ತುಂಬಾ ಎನ್ವಿರಾನ್ಮೆಂಟ್ ಹಾಳಾಗ್ತಿದೆ. ಅದರಿಂದ
ಫ್ರೂಟ್ಸ್ ತಿನ್ಲಿಕ್ಕೆ ಕಾರಣಾಗ್ತದೆ.
ಫ್ರೂಟ್ಸ್ ಅಲ್ಲಿಂದ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್
ಮಾಡೋಕೆ ಕಂಪ್ಲೀಟ್ ಪ್ಯಾಕೇಜ್ ಎವ್ರಿಥಿಂಗ್. ನೀನು ನೋಡಿದ್ರೆ ಔಟ್ಸೈಡ್ ಏನು ಫುಡ್ ಬರಲೇಬಾರ್ದು, ವೆಜಿಟೇಬಲ್ಸ್ ಅಥವಾ ಏನು ಬರಲೇಬಾರ್ದು. In this area we can't do anything, right? We need fruits, we need apple, we need so many things. Okay, so then taking that traveling from so long distance, you feel that that is going to the waste of the energy or more environmental policies to be there. So what's the use?
ಸರ್ ಆಪಲ್ ದೂರದಿಂದ ಬರುತ್ತೆ ನಮ್ಗೆ ತಿನ್ಬೇಕಾದ್ರೆ ನಾವು ಅದನ್ನೇ ನ್ಯೂಟ್ರಿಯೆಂಟ್ ಇರೋ ಬೇರೆ ಫ್ರೂಟ್ ನ ತಿನ್ಬೋದು, ವೆಜಿಟೇಬಲ್ಸ್
ಆಪಲ್ ಗ್ರೋ ಮಾಡೋರಲ್ಲಿ ಹೋಗ್ಬೇಕು. ಅವ್ರು ಗ್ರೋ ಮಾಡಿದ್ದು ಜಾಸ್ತಿ ರಿಕ್ವೈರ್ಮೆಂಟ್ ಜಾಸ್ತಿ ಇರ್ತದೆ. ಅವ್ರಿಗೆ ಏನ್ ಮಾಡ್ಬೇಕು ಅವಾಗ? ಈಗ ನನ್ನ ಊರಿದ್ದಾಗೇನೆ all the vegetables, most of the vegetables are coming from the Chickmangalore or Bangalore or some Havan like that. It's going to come. Right? We cannot able to grow anything over here because of this environmental condition. Then what we have to eat? What about rice? What about dal? What about foods? So we cannot eat anything. Right? ಈಗ ಬೇರೆ ಬರಲೇಬಾರ್ದು, ನೀರು ಬೇರೆ ತಗೊಂಡು ಎಷ್ಟೊಂದು ತಿನ್ನೋಕೆ ಸಾಧ್ಯ ಇದೆ. ಅವಾಗ ಈಗ different it's not only vegetable, there are different calories are there and vitamins are there. So we have to go through that because all the vitamins will not be the same from all the vegetables or the food. This is different. Correct? So the immediate lesson is apple energy consumption due to transportation, since it is very high right now. First of all, reduce the consumption of apple as you rightly told. Compensate it with other local fruits. And secondly, try to encourage local cultivation of the fruits which can compensate for a loss of food. Thank you. Achari, Gopika Yan, and Chandana J. Represent. Please come. Take it, sir. Introduce yourself.
Good morning, my name is present here. Myself, Kritika Chari, with my team member, Rupika and Chandana J. We are from Biodiversity Primary and High School. We thank Commander present here to giving this opportunity to present our study assessment of drinking water quality and its impact on health. Potable water means water that is safe for human consumption, meaning it is suitable for drinking. It is used in various domestic and industrial applications without causing harm to health. To be considered as potable water must meet specific quality and safety standards established by regulatory authorities such as World Health Organization, WHO, or Bureau of India standards based in India. The primary standard related to potable water in India is IIS 10500-2012, which specifies the permissible limits for various parameters in drinking water. The characteristics of potable water include taste and odor, chemical composition, microbiological safety, water treatment processes including filtration, disinfection, and sometimes advanced treatment like reverse osmosis. Regular monitoring and testing are also crucial to ensure water safe for consumption, as lack of accuracy can cause health problems and waterborne diseases. Our aim is to determine TDS, EC, and drinking water pH in the households who are using water purifiers. A survey to understand the health issues of the people in Weaver's colony, linkages between water quality and its impact on health, giving awareness and conservation of potable water is essential for health.
Our study area is selected 56 houses in River Colony, Gotigere, Badraghatta Post, Bangalore 560083, Karnataka, India. Our study site is randomly selected 56 houses in River Colony where using water purifier. We use materials in our project are TDS meter, EC meter, pH meter, various containers, notebook, pens, and questionnaires. We collect voltage numbers from the houses using clean container, measure TDS, EC, and pH. Using the calibrator TDS meter, EC meter, and pH meter, record all measurements and observation in a notebook. A questionnaire of 14 questions was prepared to collect data from residents of Weaver's Colony, household 56 samples. We visit selected 56 households in Weaver's Colony and collect water samples from each household.
Data analysis: TDS below 50 mg per liter were found in 27 samples. TDS above 200 mg per liter were found in 29 samples. EC below 0.33 microsiemens per centimeter were found in 14 samples. EC above 0.77 microsiemens per centimeter were found in 42 samples. pH below 6.5 were found in 22 samples. pH above 8.5 were found in 31 samples. samples.
This is our questionnaire of project to collect the data. I will explain the one question. Does anyone in your house suffering from BP, kidney problem, cardiovascular problem, gastrointestinal disease, diarrhea, bones problem, dental caries, respiratory issues, etc.?
Our survey report are tank water total losses 32, health problem 23, public water total losses 11, health problem 6, tubewell water total losses 11, health problem 4, well water total losses 2, health problem 1. These are graphical representation of health issues found to people using only RO, UV, RO plus UV.
Linkages between water quality and health issues. High TDS level and health problems. 29 samples at TDS level above 200 milligrams per liter. Potential health issues. Bones problem 18 cases. Dental caries 7 cases. High EC level and skin condition. 42 samples at EC level above 0.77 microsiemens per centimeter. Potential health issues. Skin rashes, dryness and allergy. 14 cases. Improper pH level and gastrointestinal issues. Many water samples at pH level outside the optimal range 6.5 to 8.5. Potential health issues. Gastrointestinal irritation. One report case of diarrhea. Source of water and prevalence of health issues. Tank water 20 out of 32 households. Public water 6 out of 11 households. Tubewell water 4 out of 11 households. Well water 1 out of 2 households reported the issues. Tank water was the liquid to the highest risk of health problem. The data highlights the impact of water quality on health and the variability of health risk associated with different water sources.
These are our awareness gallery. Now I am concluding my study. The study highlights significant variability in water quality, with many samples not meeting the best standard for potable water. High TDS and DEC levels as well as pH values outside the optimal range were common. These water quality issues correlate with various health problems reported by the residents, particularly those using tank water. The findings underscore the need for improved water treatment and monitoring system to ensure safe drinking water. Moreover, there is a crucial need for improved water treatment and monitoring system and raising awareness about water quality and its impact on health. Alongside conservation efforts to provide consistent access to potable water. Water quality is linked to physical, mental, emotional and socio-economic and community well-being. Ensuring clean and safe drinking water is essential for enhancing the quality of life. I extend our heartfelt gratitude to one and all whose unwavering support made us in Weavers Colony, especially our dedicated team members, Hasha S and Monish Raghavji, scientists, research groups, energy and wetland research groups, Indian Institute of Science and FERNSS Bangalore. Special thanks to Sri Vijla sir, Sri Kaushik sir and Sri Vinay sir for their expertise and recommendations. Appreciations for support from the school HM and guide teachers. These are our reference we took for our project. Thank you.
You have done excellent work in analyzing the problem of drinking water in your area. Don't you think it is important also to tell people how to improve the quality of the water? Another problem of invertebrates. That goes through drinks, tank water, well water, two well water, etc. How to improve the quality of the drinking water?
So we can tell for them also purify in or wash.
So that's the reason they take an RO water. They take an RO filter water and that's what they are analysis, right?
Sir, only for filter users.
No, I think they are using water filters only.
Okay, I have to think about that for your future project. You analyze the problem, explain it, and we can gradually do it. But you must go, the next step is to tell the people how to improve the quality of the water they are drinking. Okay?
Thanks.
Next presentation is Muhtaj PS, Saraswati Yadav, and Shashank PS.
Ready? Ready? Okay, start.
Good morning, honorable present here. Myself, the shares with my teammates Saraswati Yadav R and Shashank S. We are from Vijayanjali Primary and High School, Bangalore. I thank honorable present here for giving such an opportunity to present my study on ordinates of Biodiversity. Ordinates, which include dragonflies and dancerflies, are fascinating insects known for their long body, four wings and strong jaws, allowing them to capture and prey on other insects of similar size. The vibrant and algae captures closely tied to aquatic environment throughout their life cycles. At Biodiversity, these insects play a vital role in global ecosystem as they lay their eggs in or around the water with their larvae, developing aquatic environment. The sign of dragonflies and dancerflies around lake is sign of healthy interaction between lake and surrounding biodiversity. As observed in slide, these insects are in the final stage of its life, which lasts only 3 to 4 weeks. During this, primary activity is on feeding and breeding. Dragonflies and dancerflies belong to order Ordinates and distinguished by their flexible eggs, equal size wings, and vibrant body coloration, making them an integral and colorful part of Biodiversity lake ecosystem. Ordinates play a vital role in ecosystem of Biodiversity lake. As predators, they help to control population of smaller insects, including mosquitoes. Their aquatic larvae contribute to controlling aquatic invertebrates. are documentation of odorites at Biodiversity Lake, creating awareness for the conservation of odorites at Biodiversity Lake.
Stop.
Our study area is Invertebrate, Bangalore, so Jigme Hobby, Bangalore 560083. This is our this is our map. This is our starting point and ending point.
Biodiversity Lake provides an ideal habitat for odorites, supporting their life cycle and ecological role. The lake is rich in mosquitoes, flies and other small insects, offering ample prey for odorites. Odorites are most active in warm, sunny days with low wind speeds, which are optimal for their flight and hunting. Submerged, emergent and floating plants offer breeding sites where odorites lay eggs and larvae develop. Mud flats, rocks and surrounding grasslands offer shelters and breeding spots for adult odorites.
We used notebook and pen, smartphone for photography, field guide.
Our selected study area is around 1.5 km. Visited the study area every Sunday from 8 am to 10 am. From June, July, September 2024, conducted the survey in Biodiversity Lake to identify and record odorites, took photographs of odorites where odorites feed and lay eggs, recorded data and analyzed the data.
Dragonflies and dancerflies were observed primarily near the lake during morning hours 8 am to 10 am. Consistent weather conditions, cloudy with a temperature around 21 degrees Celsius, were noted during the observation period. This is our checklist of dragonflies: common laptail, Gompus vulgatissimus, Gompus. This is our checklist of dancerflies: common bluetail, Ichilura elegans, Coena gravidae. These are the pictures of odorites drawn by us, stitched well and scarlet bluetail. This is our photo gallery: scarlet steamer, wandering rider. The study period from June to September 2024 at Biodiversity Lake provided a comprehensive insight into diversity and behavior of odorites in the area. The study identified 13 species of dragonflies and 4 species of dancerflies, highlighting lake richness and diverse ecosystem. Among dragonflies, the family Regularly was the most prominent, with species wandering rider, scarlet steamer and future marsupial were frequently observed. These species are typically found around water bodies, indicating that Biodiversity Lake provides an ideal habitat with sufficient prey and breeding sites. The presence of less common species such as common laptail and Indian grasshoppers suggests that lake offers a range of microhabitats and supporting variety of odorate life cycles and feeding requirements. The dancerfly is the family of Coena gravidae with species pygmy target and common bluetail. Their abundance and diversity at this site indicates that the lake aquatic vegetation is healthy, providing suitable egg-laying and larvae development sites.
An awareness program was conducted around lake to educate local community about the importance of odorites and their role in the ecosystem. Odorites play a crucial role in maintaining ecological balance. They are effectively natural pest controllers, feeding on mosquitoes and other small insects, thus reducing the risk of vector-borne diseases such as malaria and dengue fever. Furthermore, their presence in healthy numbers indicate good water quality, which is vital for human consumption and agricultural activity. These are the pictures awareness given in our school. The documentation of odorites at Biodiversity Lake underscores the importance of wetland as a biodiversity hotspot within an urban settings. The presence of variety of 13 dragonflies and 4 dancerflies highlights the significance of the lake. We thank our HM and guide teachers for allowing us to conduct the study. We also thank Sri Vijulal sir and Sri Chaturved sir for guiding us. Our gratitude extends to scientists, field researchers, Energy and Wetland Research Group, Center for Ecology Science, Indian Institute of Science and FERNSS Bangalore. We referred Subramanian K.
IISC 1.0 2005 Dragonflies and Access Life of Peninsular India. Project Landscape Series Editor Madhav Gargil, www.wikipedia.org. Thank you.
Students have done excellent work.
Thank you, sir.
Right. The question is, what steps are to be taken to conserve these wilderness? In a way, when you talk to the villagers, if you want to talk to the villagers about the importance of wilderness, what was their reaction? First question. Secondly, what measures you suggest for the conservation of the wilderness?
We suggest that we should not pollute the lake by throwing garbages and all.
Is it number of pounds is increased because you are checking from how many years you have did this? Okay, 1 year,
4 months.
4 months, right? You referred some of the papers and all on this, right?
Yes, sir.
Is that number of this species is increases or it is going to decrease?
Sir, it
Because
is,
year to year the number is going to increase or it is going to decrease.
sir, it is increasing from August.
Increasing. To solve this what you have to do. The growth of the number of these things are we have to solve, because if you are going to allow them to grow more numbers, so that is problem to the society, right? The environment conditions. So we have to just solve this. Is it possible to reduce that? ಇದು ಸಂತತಿ ಕಡಿಮೆ ಮಾಡುವುದಾ?
No, sir, because they are very important to aquatic environment.
This is not good harmful to the environment,
No, sir.
not like mosquitoes and all.
Sir, it eats mosquitoes, flies, and control pests.
Thank you.
Next, Halima Havan, Harsha S, and Jyotika Emma.
Good morning to one and all present here. Myself Halima Hanan with my team member Harsha S and Jyotika Emma. We are from Bidanjali Primary and High School. We thank one and all present here for giving this opportunity to present this study, Survival Aquatic Macroinvertebrate Diversity and Water Quality in Bidanjali Lake. Bidanjali Lake is a key freshwater resource supports an ecosystem that plays a significant role in the well-being of surrounding communities. Among the various life forms inhabiting this ecosystem, macroinvertebrates stand out due to their vital contribution to aquatic health and their usefulness as bioindicators of environmental quality. Macroinvertebrates are crucial to the lake's food web. They contribute to nutrition cycling as they serve as the breakdown organic matter, fish and other wildlife. Their presence and diversity often reflect the ecological health and indicators of environmental quality. To identify and document the different species of macroinvertebrates in Bidanjali Lake, to assess the physical and chemical parameter water quality in Bidanjali Lake, to create awareness in Bidanjali Lake of Jigni, Hooghly, Bangalore 560083. These are our study sites and that are our study map.
Macroinvertebrates were identified from 3 designated points around Biodiversity, southwest, northwest, and west. Sampling was done within 3-meter radius from the shore and at the depth of approximately 2 feet. The study was conducted in June to September 2024, morning visit 8 a.m. to 10 a.m. Weight of the sample containers, plankton net, microscope, a field guide to aquatic invertebrates, macroinvertebrates, center, environment education, notebook, pen, smartphone to taking photographs, water sampling kit. The material that used to our study are TDS meter, EC meter, pH meter, BOD photo 125 milliliter, measuring cylinder, conical flask, pipette, burette, and chemical as prescribed in ENVI technical report 77. Various containers, labels, collected water from Biodiversity lake, plankton net, microscope, notebook, pen, and smartphone. Water samples were collected from 3 designated points, sources using 3 containers, and labeled with the sources, date, and time of collection. Using a calibrated TDS meter, the probe was submerged into a water sample. The reading was allowed to the stabilizer, and the TDS value was recorded in milligram per milliliter. We follow the same method for EC and pH meter. Measurement of DO and BOD use Fingerless method. All measurement and observation, including sample identified and test results were recorded in a notebook. These are our microinvertebrates identification. These are our gallery.
These are our identified planktons in Biodiversity lake. A survey related a rich variety of 7 species of macroinvertebrates in Biodiversity lake. The species plays a crucial role in maintaining ecological balance of the lake. The presence of dragonflies and concentrates serves as an indicator of ecosystem. The finding point to a generally healthy ecosystem, which is important for biodiversity and nutrient cycle. This is our checklist of water quality assessment. A suitable temperature indicates a suitable conditions for aquatic life. Low EC value suggests minimal ion concentration added for many freshwater species. The measured value indicates generally a clean water. pH value fall within a suitable range. Nutrients essential for plants. Macroinvertebrates are sensitive to change in the water quality and environmental conditions. Their presence, diversity serves as an indicator of the ecological health of the lake, identify pollution or habitat degradation. Macroinvertebrates are primary food sources for various species including fish, birds, and other wildlife. The local food web and promoting higher trophic levels. This is our awareness photos we are displaying in our local residents around Biodiversity lake. They are informed about macroinvertebrates play in keeping the lake and environment clean. We give some information to them like reducing chemicals runoff and stop entering the lake to untreated sewage into lake. This is our awareness photo in school.
sampling site. Well, inlet 1 and inlet 2 shows moderate levels of organic pollution and nutrient loading. Outlet 1 shows more significant pollution with BOD, low dissolved oxygen and high phosphate level. The study of macroinvertebrates in Biodiversity laid support a diverse range of sensitive species such as Dragonfly and Dragonfly nymphs, suggests that the lakes maintain a good quality of water. We extend our heartfelt gratitude to our head mister for the opportunity to conduct this study. We thank our guide teacher for their invaluable guidance throughout the process. We appreciate Rijul also for his valuable suggestion in every step of the study. We also grateful to Energy Wetland Research Group, Central Ecology Science and Indian Institute of Science Congress, Bangalore for their guidance and for providing the opportunity to carry out this research. And this our reference used in our studies. Thank you.
You mentioned about BOD in your paper. What do you mean by BOD?
Sir, biological oxygen demand.
What does it mean? What do you understand by BOD? Later. BOD. Measurement demand BOD. Use a rigorous method. What do you understand by BOD? What is BOD? You have understood BOD and then only you wanted to measure the BOD of that lake. Have you understood the concept of BOD? No. Okay, it's all right. No problem. And you talk to the people about this microinvertebrate, importance of microinvertebrates in the lake ecosystem. What was their reaction? How did they respond?
They don't know what happened.
Sir, can we answer in Kannada, sir?
Okay.
Sir, ಅವ್ರಿಗೆ ಗೊತ್ತಿದ್ದಿಲ್ಲ ನಾವು macroinvertebrates ಅಂದ್ರೆ ಏನು ಅಂತ. ನಾವು ಅವರಿಗೆ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಿದ್ವಿ macroinvertebrates ಏನಂತ. ಅವರು ಈ ತರ ಕ್ವಶನ್ಸ್ ಕೇಳಿದ್ರು, ಅವರಿಗೆ ಬ್ಯಾಕ್ ಬೋನ್ಸ್ ಇರುತ್ತಾ ಅಂತ. ನಾವು macroinvertebrates aquatic insects, ಅವ್ರಿಗೆ ಬ್ಯಾಕ್ ಬೋನ್ಸ್ ಏನಿರಲ್ಲ ಅಂತ ಅವ್ರಿಗೆಲ್ಲ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಿದ್ವಿ. ಅದಕ್ಕೆ ಗೊತ್ತಾಯ್ತಾ ಅಂತ ಅವ್ರು ನಮಗೆ ಥ್ಯಾಂಕ್ಸ್ ಹೇಳಿದ್ರು, ನಿಮ್ಮಿಂದ ನಮಗೆ ಇದು ಗೊತ್ತಾಯ್ತು ಅಂತ.
ಇಂಪಾರ್ಟೆಂಟ್ ಅಂತ ಹೇಳಿದ್ರ.
ಎಸ್ ಸರ್.
ವೆರಿ ಗುಡ್. ಸೊ ನೀವು ಸ್ಟಡಿ ಮಾಡಿದಾಗ ಬರೀ ಎಜುಕೇಟೆಡ್ ಆಡಿಯನ್ಸ್ಗೆ ಹೇಳೋದು ಒಂದು ಪಾರ್ಟ್, ಆದರೆ ಲೋಕಲ್ ಪೀಪಲ್ನ ಎಜುಕೇಟ್ ಮಾಡೋದು, ಅವ್ರಿಗೆ ಅರ್ಥವಾಗುವಾಗ ಈ ಕಾನ್ಸೆಪ್ಟ್ಗಳನ್ನೆಲ್ಲ ಹೇಳೋದು ವೆರಿ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಪಾರ್ಟ್ ಆಫ್ ಯುವರ್ ಲೈಫ್.
Thank you, sir.
Next, Miriam, Chitra, D, and Deeksha D.
We thank Corridor present here for giving us an opportunity to present our study, Survey of Macrophytes and Water Quality in Biodiversity Lake. Biodiversity Lake is geographical coordinates of a study site. Macrophytes were identified from 3 designated sources around the lake: southwest, northwest, and west, within a 3-meter radius from the shore and at a depth of approximately 2 feet. The study was conducted during June, July, August, and September. We used TVS meter, electrical conductivity meter, pH meter, collected water samples from Biodiversity Lake, notebook for recording observations and data, smartphones for capturing pictures of macrophytes in study area. We collected water samples from Biodiversity Lake using clean containers, labeled containers including the source, date, and time of collection. Using a calibrated TVS meter, we submerged the probe into a water sample and we waited for the reading to stabilize and recorded the TDS value in milligram per liter. We used that same method for EC meter and pH meter, and we recorded all measurements and observations in a notebook including the sample identifiers and test results. Macrophytes found in checklist of Biodiversity. Chala, Hydrola, Alpha Netta, Polygamo, Sparagonum, Hipomoea aquatica, water lily. These are the macrophytes found in Biodiversity. Chala, Alpha Netta, Polygamo, Hipomoea aquatica, Hydrola, Sparagonum, and Hypomoea. This is the physical parameter of our study. Example, in type 1, temperature is 26.9. EC meter in EC level is 0.73 and TDS meter level is 369 and pH level is 7.9. These are the gallery of our studies. The stable temperature at Biodiversity Lake help plants like Hydrilla verticillata and water lily grows, which cools the area and reduces water loss. The interaction between physical parameters and macrophytes at Biodiversity Lake not only supporting a thriving ecosystem but also has significant implication for human well-being. Wetland like Biodiversity Lake provide a crucial ecosystem directly benefiting to the local community. Various macrophytes including Chara, Altalentra, Spalaneum and water lily were present in June, July, August and September. Due to the overflow, we couldn't find Polyganium Hipomoea aquatica in August, and in September, Hydrilla verticillata was not visible. Hydrilla verticillata water lily were found throughout the lake. Water quality measurement, temperature, electrical conductivity, total dissolved solid were consistent across the lake. Critical effect to clean and conserve the lake are promising. The lake remains vital habitat for various aquatic species. Macrophytes at Biodiversity Lake serve as bioindicator of the water quality and ecological health. Hydrilla verticillata suggesting a balanced nutrient level and good water quality. This species is known as for sensitive to change in water quality, particularly in nutrient level. Chara indicating low pollution level and stable pH, especially at inlet. Chara is a strong indicator of the low pollution level and stable pH. Water lily reflects stable, slightly alkaline pH level across the lake. These bioindicators confirm that the lake's physical parameters are optimal for diverse and balanced ecosystem. Monitoring these bioindicators confirm that the lake bioindicators is essential for detecting potential changes in water quality. Biodiversity Lake has well-balanced and healthy aquatic ecosystem. The health of macrophytes supported by these physical parameters is crucial for maintaining biodiversity and well-being of the local community. Protecting and monitoring this contribution and their impact on macrophytes is essential for the lake's continued ecological and social benefits. We thank our lecturer and guided teacher for allowing us to conduct this study. We thank Chemistry US for designing Therapy, a group member of the study, documented macrophytes, location and Biodiversity. We will also thank Sri 2016 IISC. Thank you.
So all of you have exhaustively studied it. So all of you, three, 4, 5 papers are exclusively on this lake.
Yes, sir.
And totally you have exhaustively studied all the parameters of this lake and found that it is very healthy and commendable. What are the uses of this lake for the people? ಜನರು ಈ ಕೆರೆ ನೀರನ್ನ ಯಾವ ರೀತಿ ಬಳಸ್ತಾರೆ?
Sir, fishing. Bird watching ತರ ಏನಾದ್ರು
ಫಿಶಿಂಗ್ ಬಳಸ್ತಾರೆ, ಡ್ರಿಂಕಿಂಗ್ ವಾಟರ್ ಪರ್ಪಸ್ ಬಳಸ್ತಾರೆ,
Yes, sir.
ಬಳಸ್ತಾರೆ. ಆಮೇಲೆ
Sir, making for this wash
ಜನ ಏನಂತಾರೆ? ಲೇಕ್ ಬಹಳ ಚೆನ್ನಾಗಿದೆ, ನೀರು ಕ್ಲೀನ್ ಆಗಿದೆ ಅಂತಾರ, ಅಥವಾ ಗಿಡ ಬೆಳ್ಕೊಂಡ್ಬಿಟ್ಟಿದೆ, ಏನೇನೋ ಹೋಗ್ತಾ ಇಲ್ಲ ಬರ್ತಾ ಇದೆ, ನೀವು ಎಕ್ಸಲೆಂಟ್ ಅಂತೀರಿ. ಆದರೆ ಜನಗಳು ಹಾಗೆ ಹೇಳ್ಬೇಕಾಗಿಲ್ಲಲ್ವಾ? ಸೊ ವಾಟ್ ಇಸ್ ದೇರ್ ಒಪಿನಿಯನ್?
ಸರ್ ಅವ್ರು ಹೇಳಿದ್ರೆ ಅದೇ ನೀವು ಹೇಳಿದ್ರಲ್ಲ ಸರ್, ತುಂಬಾ ಪ್ಲಾನ್ಸ್ ಎಲ್ಲ ಬೆಳ್ಕೊಂಡ್ಬಿಟ್ಟಿದೆ ಅಂತ, ಬಟ್ ಅವ್ರಿಗೆ ಗೊತ್ತಿರಲ್ಲ ಏನಕ್ಕೆ ಅದು ಬೆನಿಫಿಟ್ ಅಂತ. ನಮಗೆ ಗೊತ್ತಿದೆ, ಸೊ ಅವ್ರಿಗೆ ಅರ್ಥ ಮಾಡಿಸಿದ್ರೆ ಅವರು ಅರ್ಥ ಮಾಡ್ಕೊಳ್ತಾರೆ.
ಅರ್ಥ ಮಾಡಿಸಿದ್ರೆ
ಸರ್,
ಒಬ್ಬರ ಜೊತೆ ಮಾತಾಡಿದ್ರೆ,
ಸರ್,
ಯಾಕೆಂದ್ರೆ ಅವರ ರೆಸ್ಪಾನ್ಸ್ ಚೆನ್ನಾಗಿದೆ ಅಂದ್ರೆ
ಸರ್,
ಸ್ಟಡಿ ಏನು ಉಪಯೋಗ ಅವ್ರು ಏನೋ ಗಿಡ ಬೆಳ್ಕೊಂಡಿದ್ದೀನಿ
ಎಸ್ ಸರ್.
So educating the local people is very, very important. More important than the scientific study. Okay.
Yes, sir. Thank you, sir.
Next, Eknath GP, Jeevan M and Sanjay S.
Good morning, one and all present here. Myself, Eknath GP, is my friend Jeevan and Sanjay S. We are from Vidyajali Primary and High School. I thank one and all present here to give me this opportunity to present our study, observation and documentation of spiders in Weaver's Colony. Weaver's Colony is a semi-urban region of Bangalore with a mix of residential, commercial and open spaces with greenery patches. The colony supports a diverse of flora and fauna, including various spider species. The helping maintain a balanced ecosystem and reduce harmful pest population, which is especially beneficial in urban areas. Spiders play a crucial role as natural pest controllers by eating flies, mosquitoes, and other arthropods. Our objectives are observation and documentation of spiders in selected houses in Weaver's Colony to document identified spider species in regular intervals to conduct awareness about the importance of spider.
Our study area is Weaver's Colony, Bangalore South, 28 Post, Banapeta Road, Bangalore 560083. The selected study area is around 1 km square. And this is our study map. And this is our study space with coordinates by Jeevan M, Eknath GP, Sanjay S and Haripreet P. Our materials are spider identification guide, backyard spider book, notebook and pen for data recording, smartphone for taking photographs and recording location. 4 study sites were identified. The observation was made every Saturday and Sunday of June to September 2024, morning from 7 a.m. to 8 a.m. and evening from 5 a.m. to 6 p.m. in our study center.
This is the observation of individual key study sites of spider studies, and this is the monthly statistics. And this is our spider species documented with common scientific family those of found in June, July, August, and September, stored in Havan, Totally, we documented 20 different spider species across our first studies. And this is the checklist of spider individual of the study sites. And this is our botanic fact of the jumping spider, agricultural spider, breeding spider, common table spider, body longest spider, stellar spider, wall spider, forest omnivore spider, sun spider, aerial spider. The study documented 20 different spider species across the study sites in US Colony, indicating significant spider diversity worldwide. The first study sites and address species respectively with 8 and 9, while the 8 and 10 sites recorded each species. This suggests the certain macroinvertebrates are more suitable for spiders due to factors like vegetation, food availability, or less human disturbance. Monthly observations was the highest diversity was observed in September 12 and August 11. Species specific and some species such as smaller omnivores, stellar spider, and body longest spider were present in multiple months. Other like marble omnivore, yellow sword spider were observed in only specific months. This might indicate a seasonal difference. The presence of diverse families such as Algae, Invertebrate, and Oxycrine studies suggest the healthy and balanced ecosystem in US Colony. The study highlights the ecological richness of US Colony, making it a potential habitat for various arthropods.
Spider play a crucial role of biocontrol, natural pest controls by feeding on insects such as birds, flies, and other small arthropods, regulating the pest population and reducing the usage of chemical pesticides. Spider contribute in maintaining a ecological balance and support biodiversity and in reverse colony. Spider is a
You can just look the board, no issue.
the variety of family observed like omnivore spiders such as forest omnivore and marble omnivore, suggesting a highlighting availability of prey and the presence of vegetation which provides the suitable vegetation for the breeding. We gave awareness to people of Biodiversity, raising awareness about ecological role of spiders in vital community of Biodiversity. In our study, findings show that spiders are not only harmless, they are beneficial in biocontrol by reducing the pest population. This awareness can lead to improve the coexistence where the spiders are appreciated for the contribution of maintaining the ecological balance and health. This is the awareness we gave for the people of Biodiversity. This is the awareness we gave for the children of our school.
Spiders play a crucial role as natural pest controllers and contribute to a biodiversity in the ecosystem, which supports their well-being by reducing pests. A total of 20 different spider species recorded in our US Colony across 4 study sites. This emphasizes the spiders are well adapted to
ನಿಮ್ಮಲ್ಲಿ ಒಂದು ಕಡೆ ಮ್ಯಾಕ್ಸಿಮಮ್ ಅಬ್ಸರ್ವೇಷನ್ ಸೆಪ್ಟೆಂಬರ್ ಅಂಡ್ ಅಕ್ಟೋಬರ್ ಪೀರಿಯಡ್ ಅಲ್ಲಿ ಮ್ಯಾಕ್ಸಿಮಮ್ ಅಬ್ಸರ್ವೇಷನ್ ನೋಡಿದ್ವಿ ಬರ್ತಿದ್ವಿ. ಯಾಕೆ ಸೆಪ್ಟೆಂಬರ್ ಅಕ್ಟೋಬರ್ ಮೇಲೆ ಮ್ಯಾಕ್ಸಿಮಮ್ ಅಬ್ಸರ್ವೇಷನ್?
ಸ್ಪೈಡರ್ಸ್ ಸನ್ಲೈಟ್ ಆಕ್ಟಿವ್ ಆಗಿರುತ್ತೆ. ನಾವು ಸ್ಟಡಿ ಫಸ್ಟ್ ನಾವು ಲಾಭವಾಗಿ ಡೇಟು ವೆದರ್ ಕಂಡೀಷನ್ ನೋಡ್ತೀವಿ. ಮತ್ತೆ ನಾವು ಜಾಸ್ತಿ ಕ್ಲೌಡಿ ಆ ಥರ ಸ್ಪೈಡರ್ ಹೆಚ್ಚಾಗಿ ಕಾಣಿಸಲ್ಲ. ಸೊ ಆದಷ್ಟು ಸೆಪ್ಟೆಂಬರ್ ಸ್ವಲ್ಪ ಸೀಸನ್ ಚೇಂಜಸ್ ಆಗಿದೆ. ಆಗ ನಮಗೆ ಸ್ವಲ್ಪ ಸನ್ಲೈಟ್ ಸೊ ಆಗಿ ನಮಗೆ ಸ್ಪೈಡರ್ಸ್ ಹೆಚ್ಚಾಗಿ ಕಾಣಿಸ್ಬೋದು.
ಅದರ ಬ್ರೀಡಿಂಗ್ ಸೀಸನ್ ಬ್ರೀಡಿಂಗ್ ಸ್ಪೈಡರ್ಸ್
ಜಾಸ್ತಿ ಸಮರ್ ಟೈಮಲ್ಲಿ
Next, Sanjana K, Viman Raj, and Manoj Jha.
ಒಂದು ಸೊಲ್ಯೂಷನ್ ಇದೆ. ನೀವು ನಂತರ ತಪ್ಪದೆ ಕಲ್ಪನೆ ಬರಬಾರದು. ನೀವು ಬಹಳ ಒಂದು ಆತ್ಮೀಯವಾದ ಸೊಲ್ಯೂಷನ್ ನೋಡಿದ್ದೇನೆ. ನೀವೆಲ್ಲ ಕಾಲೇಜ್ನೆಲ್ಲ ನೀವು ರೆಕಾರ್ಡ್ ಬಂದಿದ್ದೀರಿ. ರೆಕಾರ್ಡ್ ಮಾಡೋದಂತ ಇಲ್ಲ. ನಿಮ್ಗೆ ಯಾವಾಗ ಒಂದು ರೆವಿಷನ್ ಸ್ಟೇಟ್ ಬರ್ತದೆ ಯಾವಾಗ ನಿಮಗೆ ಅಲ್ಲಿ ಕೆಲಸದಲ್ಲಿ ಬಿಡಿ. ಈ ಕೊಟ್ಟಿನ
No problem.
and good feeling confusion. Okay? That is.
Good morning to all our present here. Myself Sanjana K with my team members Manoj M and Vivan Raj S. We are from Vidyajali Primary and High School. We thank all our present here for giving this wonderful opportunity to present our study observation of moth using light here. Biodiversity is a notable aspect that supports a diverse array of species which play a crucial role in the local ecosystem. Moths are nocturnal lepidopteran insects characterized by their feathery antenna, robust bodies, typically duller coloration compared to butterflies. The plant-eating caterpillars contribute significantly with the ecological balance within this urban environment. As essential for primary pollinators, moths fill the gap left over by diurnal pollinators by transporting large amount of pollen grains over long distances. Additionally, moths and their caterpillars serve as key food sources for numerous species including bats, birds and other predators. Our study objectives are observation and documentation of moth diversity in beavers colony, to conduct awareness about the importance of moth and to educate people in beavers colony.
Our study is given for the Bengaluru sir, Kotigere post Havan, Bengaluru 560083. Our study set is practical geographical info about the Shanghai Beavers Colony, Kotigere, Bengaluru, and this is our coordinates documented by India, Vivan Raj, Manoj Sanjana.
A light lab was used to observe and document the moth diversity in beavers colony. Light lab was activated at 6 p.m. to 6 a.m. in the selected residences with a wide thin cloth measuring 3 feet
members with around Moth tribe. Checklist of problem must features to identify monthly in diverse colony. Moth species development monthly in various statistics of diverse colony.
Checklist of Moth species from the settlement. Right here we have global Moth family form. This is our Moth species. This is our Moth family by chart. Photography of Moth documented by Havan, by Macroinvertebrates, by Dragonfly, by Biodiversity, by ecological. The biodiversity study in diverse colony and biology nature will document different species of Moth from June to September. July and August diversity of Moth with 36 species, followed by August and September will document 34 species of Moth. June and August diversity of Moth with 28 species. June is after time of observation from June 18. Length of South diverse colony and the highest diversity of Moth species with up to 9 species observed in June to September. The ideal range of Julia and Peninsula was the most frequently observed species in our state. The combination includes Moth activity, particularly and sunny weather. Moth includes sightings while dry and sunny spells trigger fear sighting. The Indian greenhouse Gloria and Peninsula, big by Thomas, Gloria rectangular and Nolan transforma were the most frequently observed species in our state. Species such as common plasma, Erodactyla, rectangular greenhouse and Moth were observed regularly in July. The listed species are present.
Moths play important role in diverse colony in ecosystem. Moths pollinate flowers at night, similar how bees and butterflies do during the day, helping plants to grow and produce seeds. Moths and their caterpillars serve as key food sources for birds, bats, and other animals, making moths an important part of the food chain. Moths helping break out of their plants and leaves, enriching the soil and supporting new plant growth, making them valuable to the urban ecosystem.
This is our organization gallery.
The study conducted in diverse colony Bengaluru demonstrates that these urban bats supersede diverse array of moth species. We documented 64 species of moth during June to September 2024. The findings highlight the crucial role of moth serving as nighttime pollinators, food source for various predators, contributing to nutrient cycling. The documentation of moth across different study sites shows variations in diversity. The study emphasizes the importance of conserving moth habitats in urban areas like diverse colony, protecting green spaces, and raising awareness to maintain biodiversity. We thank our headmisters and guide teachers for allowing to conduct our study. We appreciate all our team members who created environment for moths in their houses and documented them. We extend our gratitude to Energy and Wetland Research Group, Center for Ecology Science, Indian Institute of Science, Bangalore, as well as Sri Vigilance and Sri Chetras and Asar for their guidance. Our gratitude extends to scientists, field researchers, and experts of the Energy and Wetland Research Group, Center for Ecology Sciences, Indian Institute of Science, and MERNSS Bangalore. These are our references. Moths of Bangalore run by Environmental Management and Policy Research Institute, IISC, and 2015. A brochure of
ಮಾರ್ಕ್ಸ್ ನೀವು ಬೆಳಗ್ಗೆ 5 ಗಂಟೆಯಿಂದ ರಾತ್ರಿ 6-9 ಟೈಮಿಂಗ್ ಎಲ್ಲಿ ಸ್ಟಡಿ ಮಾಡಿ ಮನೆ ಬಿಲ್ಡಿಂಗ್ ಹೊರಗಡೆನಾ
ಮನೆ ಆಚೆ
ಮನೆ ಆಚೆ ತೋಟ ಎಲ್ಲ ಇರ್ತಾರೆ ಆ ಕಡೆ ನಿಮ್ಮ ರಿವರ್ಸ್ ಫೋಟೋ ಇಲ್ಲಿನೇ ಹೋಗ್ಬೇಕು ಎಷ್ಟು ಮನೆಗಳಿದೆ ಅಂದ್ರೆ ಅಲ್ಲಿ ಫ್ರೀ ಏರಿಯಾನೇ ಕಾಣಿಸ್ತಾ ಇದೆ ನಮಗೆ. ಹಾಗಾಗಿ ಮಾರ್ಕ್ಸ್ ಎಲ್ಲಿರೋ ಜಾಗ ಇದೆ
ಸರ್. ನಾವು ಲೈಟ್ ಟ್ರ್ಯಾಕ್ ನ ಅರೇಂಜ್ ಮಾಡ್ಬಿಟ್ಟು ಅದು ನೈಟ್ ಪಾಲಿನೇಟ್ ಮಾಡ್ತಿದ್ದೇವೆ ಸರ್. ನಾವು ನೈಟ್ ಪಾಲಿನೇಟ್ ಮಾಡೋ ಫ್ಲವರ್ಸ್ ಜಾಸ್ಮಿನ್ ಅಂಡ್ ಮೂನ್ಲೈಟ್ ಫ್ಲವರ್ಸ್ ನ ಇಕ್ಕಿದೀವಿ. ಸೊ ಅದಕ್ಕೆ ಪಾಲಿನೇಟ್ ಮಾಡೋದಕ್ಕೋಸ್ಕರ ಬರುತ್ತೆ. Moths are nocturnal lepidopteran insects, but butterflies, they are diurnal pollinators. The feathery moths, feathery antenna, robust bodies, and duller coloration compared to butterflies. And butterflies will be active on days.
You have told us about two lakes.
Yes.
The first one is
Dragonfly.
is there any inflow of drainage?
So it's not a lake and two meters from sewage, but there's nearly 170 meters from water.
So that's why it's forming.
Yes, sir.
you forming in Chinnapanadiki? That's mainly due to? Mainly due to? Use of detergent. Have you told your thing? Have you talked to the local residents about the use of detergent and its impact on the lake?
Thank you, sir.
Somebody proposed
kingfisher, the oriental darter, Eurasian moon and little egret. So in this slide you can see the 200 lake species with the scientific name Macroinvertebrate, family, Havan and IUCN status. So in 200 lake there is only one clear threatened bird species, that is the painted stork. Again in this slide I have noted whether the bird species were present or absent or after my 7 days of visit. These are the pictures of the 200 lake species. This is a Venn diagram showing the specific species found in Munekala Lake that are 13 in number, that 10 species found in both the lakes that are 10 in number, and the species found only in Kumarahalli Lake that are 5 in number. So in this slide you can see the category of the major birds found in both the study areas. So the rating birds on the other hand are most in number, and the birds of prey are less. After visiting both the lakes, I can conclude that Munekala Lake has a lot more trees than Kumarahalli Lake. So because birds need trees for their nests, food and refuge, Munekala Lake is greener, so the diversity of birds is more there. As I said earlier, 30 species have been specifically spotted in Munekala Lake, 5 in Kumarahalli Lake, and 10 common in both the lakes. So the total number of species is 37. So it is our duty to preserve wetlands like Munekala Lake as they support bird diversity by providing them food like invertebrates, plant materials and abundance of fish, and they also provide a nesting sites and refuge for the birds. So these are the ways we can improve the diversity of birds. Preserving the bird habitats that are already there and planting more trees, especially the fruit-bearing trees, so they supply the birds with food throughout the year, and avoiding the use of chemicals that can harm the birds directly or indirectly, placing decals or ceilings on windows to prevent the birds from colliding with them, and educating people about the importance of wild bird populations. The references I used are listed in this slide. I thank IISC for giving me this opportunity, and I thank Research and Development Department of my school for guiding me throughout this project. Thank you.
What are the seasons in which you have observed larger number of birds?
July and August.
It's a rainy season. What about in the summer season?
Summer season, the water in the lake was less, so I saw less wading birds and the rail birds. I saw more of the raptor birds that are the birds of prey.
What do you think? The birds will stay in one place all the time, or they will go wherever the food they will find, will go and come back again? Do you think?
Yes. So if they don't find enough food in one lake, they might go visit some other lake and find their food.
Okay, thank you very much.
Students, we have
us. You know that our students should always coordinate.
lunch break, whatever the time is allotted, please make sure you are on time. Like you come back on time, okay? We are starting the session now. So the next presentation is from, please, Algae Biodiversity Institution, Sai Prasad. Yeah, please start with your presentation.
Good morning to all the judges, my dear fellow friends, and all the teachers. I am Sai Prasad, and this is my presentation. We are from grade 8, Biodiversity Biological Comparative, and our research title is Contaminating Waters, an ecological study of the Macroinvertebrates. Dragonfly, located near the heart of the city Comparative, is one of the city's largest lakes. It was a catchment area made alongside the Ganga River for irrigation purposes. The Water Lake, too, was made for the same purpose, and the first is both these lakes in the recent past have been subjected to heavy pollution. Our main objectives with this research includes finding the flora around the lake and finding how the water quality and the soil quality are affected. There is a lot of importance for mitigation in urban areas, which include carbon sequestration, temperature regulation, and air quality control.
So our study area includes Bellu Lake, Vattu Lake, and the stream which connects both the lakes. Bellu Lake is the largest lake in Bangalore and it's subjected to pollution as 60% of Bangalore's untreated sewage water enters the lake. Vattu Lake too is subjected to pollution as the water from Bellu Lake flows into the Vattu Lake. Bellu Lake has caught on fire in 2017, which was in 2017, and Vattu Lake too was famous for the foaming incident which again took place in 2017. So this is the map of the river which flows through both the lakes. You can see the Bellu Lake and Vattu Lake in the river. So this is the satellite image which was taken in 2009. Here we can see that there is a lot of water in the lake and most of the area around the lake is farmland and there are not much buildings. This is the image which was taken in January 2020. Here we can see that the water content has reduced and most of the farmlands have been enclosed for breeding purposes. So this is the current image of the lakes. Here you can see there is not much water. This is because the lakes are under rejuvenation by the breeding. So we had collected our samples on 27/6/2024, 25/8/2024 and 8/9/2024. Our soil samples were collected at an average depth of 15 centimeters. To identify the plant species, we used Google Lens to capture the pictures and search for its scientific name and common name. Whereas for the local name, we asked the local name.
Through this method, we found 3 types of vegetations in these water bodies. They include aquatic vegetation, migratory vegetation, and terrestrial vegetation.
So the aquatic vegetation in Vattu Lake includes water hyacinth. For riparian vegetation includes river tamarind, castor plant and monophase. And the terrestrial vegetation includes African tulip tree and Cascadian pine. For Bellu Lake, for aquatic vegetation we found water hyacinth. Riparian vegetation we found Saccharina, river tamarind and castor plant. And for terrestrial vegetation we found
Tree vegetation was mainly dominated by riparian species, which include the morning glory, Tasmina plant, black crocus, and the Mediterranean lantern. These are some of the images of the vegetation which we took. We had used the Jalna water testing kit for testing our water samples and the soil testing kit for our soil samples. The reagents we used include magnesium sulfate, alkaline calcium iodide, phosphoric acid, and sodium thiosulfate for the soil oxygen, whereas for ammonia test we are using NaCl reagent. For the phosphorus test we are using ammonium molecule and stannous chloride. For pH we are using vector, chlorine sulfur, pH vector, and thermometer tubes for the thermometer testing. For the soil test we are using pH reagent 1 and 2, nitrogen reagent 1 and 2, phosphorus reagent 1 and 2, and potassium reagent 1 and 2 along with TVRAMACHANDRA. These are some of the images which we took while conducting the water tests, and these while conducting the soil tests. We have also calculated our results
These are the water test results in bar graph. We did DO, mixed oxygen, ammonia, phosphate, and nitrate. This is the pH of soil this is the left side, and this is the pH of water. The middle one is for turbidity of water, and the last one is for TVS of water. This is the soil test results in the bar graph.
This is an article published by the Times of India in 2017. This shows on the left side the image of Algae taken in 1989, whereas on the right side is the same image taken in 2017. It's hard to believe that Algae being such an ecological hub has now turned into a waste site. Here we have taken some images of the weed growth on the surface of Vato Lake. Sadly, Vato Lake has also fallen into the trap of rapid urbanization.
So the National Green Tribunal has ordered to halt the discharge of untreated sewage and industrial effluents into the lake. The BDA, which is the Bangalore Development Authority, has proposed several institutional plans and plans to ensure that the water entering the lake is treated to the required standards. And currently, it's taking plans to discriminate the lakes. The community members also actively participate in human rights or organized. The Vato Lake is a significant pollution which leads to nutrient overload and eutrophication. Due to nutrient overload, invasive species thrive and they threaten the native species. Water mismanagement such as improper irrigation practices and improper water diversion and excessive extraction of groundwater threaten the aquatic species, marine species, and the terrestrial species.
So, through these research, we have come to the conclusion that pollution has increased the phosphate levels and reduced the oxygen levels in the lake waters. But still, we have found some plant species such as eucalyptus, river target, water lilies, etc. The local species growth have been threatened due to high nutrient overload. We have a lot of recommendations for these states to guide. They include having adequate buffer zones, using mitigation techniques, and creating sewer treatment plants on the waste or the sewages. These are some of the references which we have used. We thank Dr. TVRAMACHANDRA sir and the IISC team for helping us through this daily symposium, and Dr. R&D department for providing us all the resources. We also thank Sindhu ma'am and Govinda sir for providing us help and guiding us through this. Thank you. Any thoughts?
Without oxygen ಅಂತ ಒಂದು ಪ್ಯಾರಾಮೀಟರ್ ಕೊಟ್ಟಿದ್ದೀರಿ.
Sir,
What's the importance of dissolved oxygen?
so dissolved oxygen is really important for aquatic life to thrive, as if there is less oxygen dissolved in the water, then there is less chances for aquatic animals to get the proper demand of oxygen, so hence they might die of suffocation.
ಇನ್ನೊಂದು ವಾಟರ್ ಹೈಸಿಂತ್ ಬೆಳೀತಾ ಇದೆ.
Yes sir.
ವಾಟರ್ ಹೈಸಿಂತ್ ಬೆಳೆಯೋದು dissolved oxygen ದೊರೆಯೋದು.
So like because of pollution only these have gone down, and water has growth has like caused other big species or that not be multiplied, as like mostly those species like very little oxygen over there.
Indian collagen, and Asian coral. Here is a chart representing the bird population in both the lakes. Here the blue represents Saki Atlantic and the orange represents Invertebrate. You can see that more birds were found in Saki Atlantic because of the reason as it was bigger and cleaner than the blue. Now, moving on to my second objective, to observe the role and significance of birds within the environment. Birds are really significant as they help in seed dispersal, aiding nutrient cycles, helping food chain as well, control insect population, and a lot more. They also help in attracting tourists, thus increasing local economy. We have seen how significant birds are, yet some of our careless actions affect them a lot. Factors such as urbanization, deforestation, and climate change alter ecosystems crucial for survival of birds. Loss of nesting sites, food sources, and migration routes can threaten the species' survival. Now, we have realized the mistake, and as a society, we have taken a few measures. Some of them include creating wildlife parks and sanctuaries, restoring natural degraded habitats such as wetlands and forests, conducting public awareness campaigns, and enforcing laws and regulations about wildlife. Conclusion: birds are extraordinary creatures. Number of birds found in Sankitan Lake was species 6 with a total of 63. Number of birds found in Blue Lake was species 7 with a total of 55. Birds have a huge significance on the economy, culture, and ecosystem of a place. Some of our careless actions affect their habitat a lot. We have realized our mistake and have taken several efforts to resolve them. Here are some of the references I have used. For Sankitan Lake, I have used one from Research Kit and the other from Keyboard. For the Blue Lake, I have also used the other one from Keyboard. I would like to express my gratitude to IISC for providing me with this opportunity. I would also like to thank the teachers of my school for encouraging me, guiding me, and supporting me. I would like to thank the R&D department of our school and our principal and chairman, sir. Thank you all.
Very good work. Very what's the problem that these birds are getting in these schools?
In Sankitan Lake, the people of the lake, they feed the birds, they feed them seeds. In Blue Lake, they just find a food that is naturally present in the ecosystem.
What they say, people feed the birds in
There are,
the water.
because in some point of view it is good, in the other it is bad. Like the birds, they are getting used to this comfort zone and they are not going out and playing, which is their normal activity.
And you mentioned that the large number of cormorants are coming to these birds.
Yes.
What's the specific reason for the increasing number of cormorants in these birds?
When we are doing these projects, you just go one day and just look around, where are the birds singing, what are they eating, and things like that, and then let it down. Or you go like frequently, like once a week, just monitor them. Okay.
I go like every once a week.
Okay, thank you.
Thank you, sir.
Very nice.
Next is Anvita Krishna.
biodiversity and the type of conservation needed in each particular place. And they're highly affected by climate changes, and so they give us ecological insights. And as we all know, birds also help the environment in several ways, such as supporting the biodiversity, keeping a balance in the ecosystem, and also promoting ecotourism and culture. So these are my study areas for the project. And the first one is Havan Wildlife Reserve. So this is a place where urbanization is nil, and Hoskote Lake is a place where urbanization is slight, and Solitary Lake is a place where urbanization is intense. So I have tried to observe the images of the birds, and I have tried to compare the birds which were in the urbanized areas, as my study areas are a wildlife reserve, an intensely urbanized area, and a slightly urbanized area. And I have visited my study areas in between the times of 6 a.m. to 8 p.m. and 4 p.m. to 6 p.m. And we have captured the pictures using the cameras film and Nikon. The specifications are given here, and the pictures used in these PPTs are clicked by my dad. And I use the night transit method for spotting the birds. So these are the birds which I spotted in Kabini, and I enjoyed watching the osprey because the osprey is a bird of prey which mainly eats fish, and so it was exciting to watch how it zoomed into the lake and caught the fishes like the kingfisher. And these are the birds which I spotted in Hoskote Lake, and I was lucky enough to spot the spot-billed pelican because the spot-billed pelican is a migratory bird, and it only migrates in the winters. And these are the birds which I spotted in Solitary, and here I would like to emphasize the grey-headed scorpion. So the grey-headed scorpion was completely developed, and it was an adult one, and the plumage of it was slightly darker than the counterparts of the Kabini scorpions. And these are the birds which I found in total, and here I have given the bird's name, the bird's scientific name, and I have color coded each of the place, which is Kabini is for green, blue one is for Hoskote, and pink is for Solitary. So I have also given the count of the bird in each of the place. And for this analysis, I have classified the plumage characteristics as bright for the ones which were easy to spot, camouflage for the ones which were completely blended into the woods, and dull for the ones which were dull in plumage, for the ones which were dull in plumage, which when compared to the parts of Kabini, and camouflage dull for the ones which were camouflaged and also dull in plumage. And I have also given the IUCN status of each bird. So the Malabar hornbill is vulnerable, which means it is at a high risk of extinction in the wild. And the oriental darter and the spot-billed pelican are near threatened, which means it is in the middle stage of low risk and high risk of extinction in the wild. And most of the birds which I saw were least concerned, which means they have no threat. So this is the paragraph which I got from the data in the previous slide. And here you can see that about 70% of the birds which I saw and the rest was made and the bird had which means that most of the birds which were seen in solitary were there. So here we can conclude with the information I got that the birds which live in urban areas are have slightly darker and duller plumage than the birds in the wildlife reserve, and the urbanized areas birds are slightly darker and duller than the birds in company because the dirt and dust present in the atmosphere is sticking to their feathers and body, so it is making it dull and darker to look at. And the birds are showing bioindicators which show the rapid and uncontrolled pollution in the environment. So I would like to recommend more and more focused analysis in each urban area to see which chemical is used, which chemical is polluting the area, and we can utilize it with the corrective measures. And these are some of the birds which I found in all over the study areas. And these are the references I used for the project. And I would like to thank the IISC for giving this great opportunity, and I would like to thank the R&D department of Comparative for giving me this opportunity, and I would like to thank my parents for their support. Thank you.
What's your conclusion? If more bright colored birds come to the lakes in the urban area, we will say that the lakes are very good. If duller colored birds come, the lakes are not good. Is that the conclusion?
Sir, it's actually like in the wildlife reserves, the dust and dirt is not that much, so it doesn't really affect the birds. And whereas in the urbanized areas, the dust and dirt is more, so it just sticks to their body.
But you must also see bright colored birds are attractive to human beings. But is it very important for the bird as such?
No, sir.
ಅಲ್ವಾ? ಬ್ರೈಟ್ ಕಲರ್ ಇದ್ರೆ ನಮಗೆ ಚೆನ್ನಾಗಿ ಕಾಣುತ್ತೆ, ಆದ್ರೆ ಪಕ್ಷಿಗೆ ಬ್ರೈಟ್ ಕಲರ್ ಇರೋದು ಒಳ್ಳೆಯದ ಇಲ್ವಾ ಅನ್ನೋದನ್ನ ಅರ್ಥ ಮಾಡ್ಬೇಕು. ಯಾಕಂದ್ರೆ ಶತ್ರುಗಳಿಗೆ ಬೇಗ ಕಾಣುತ್ತೆ ಅವರು, ಅಲ್ವಾ? ಜನಗಳಿಗೆ ಅಟ್ರಾಕ್ಟಿವ್ ಆಗಬೇಕಾದ್ರೆ ಜನಗಳು ಅದರ ಹತ್ರ ಹೆಚ್ಚಿಗೆ ಹೋಗೋದಕ್ಕೆ ಇಷ್ಟಪಡ್ತಾರೆ. ಬ್ರೈಟ್ ಕಲರ್ ಆಗಿತ್ತು ಅಂತಂದ್ರೆ ಅದಕ್ಕೆ ಈ ಕ್ಯಾಮೋಫ್ಲೇಜಿಂಗ್ ಅಂತ ಹೇಳ್ತಾರೆ, ಅದು ಸುಲಭ ಆಗ್ತದೆ. ಅದು ಯಾವ ಏರಿಯಾದಲ್ಲಿ ವಾಸ ಮಾಡುತ್ತೆ ಆ ಕಲರ್ ಇದ್ರೆ ಶತ್ರುಗಳಿಗೆ ಬೇಗ ಕಾಣೋದಿಲ್ಲ, ಬೇಗ ಮರೆ ಮಾಡೋದು ಮಜಬೂರು. ಅದರಿಂದ ಕಲರ್ ಅನ್ನೋದು ನಮ್ಮ ದೃಷ್ಟಿಯಿಂದ ನಾವು ಅಸೆಸ್ ಮಾಡಬಹುದು.
So I was just trying to find out if the birds are dull in color when we go to the side of urbanized areas, and if they are dull in the like wildlife reserves. I was just trying to find out.
This is another aspect of the problem. Keep it here.
Thank you.
Good morning everyone, I am Nishamba. This is my teammate Lakshmi. We are from Biodiversity class for Nirma class studying ingredient. We are going to present a presentation called soil analysis. Today, we will discuss the crucial role of soil plays in our environment and the alarming damage it has faced due to deforestation, overfarming and bad management. Soil is not only the foundation of agriculture, but also sustains life on earth by enabling the growth of plants, a primary source of food. As healthy soil is integral for successful plant growth, its degradation poses a significant threat to our ecosystem and food security. Let's move on to the objective. This is the location of our study area. Samples were collected from two different locations in Karnataka. Selected area provided us with diverse soil types, allowing for comprehensive comparison. These are the snapshots we captured by collecting our soil samples in our study area. First two images are from farm soil sampling area, and third and fourth images are from lake soil sampling area. For moving further in this presentation, we must We would like to thank IISC for giving this blissful opportunity for conducting our research. We would also like to thank Algae Institute for providing us this opportunity and proper patient time. These are the online websites that we took information for that project. Nali soil, Nali sugar. Let's join our hands to preserve Earth for our future. Thank you.
Soil compared. Which one is more fertile?
Farm soil.
Huh?
Farm soil.
NPK, chemical fertilizer ತುಂಬಾ ಬಳಸ್ತಾರೆ. Chemical fertilizer ಅಂದ್ರೆ NPK, nitrogen, phosphorus, and potash. So ಇದರಿಂದ soil quality ಜಾಸ್ತಿ improve ಆಗ್ತಾ ಇದೆಯಾ ಅಥವಾ ಕಡಿಮೆ ಆಗ್ತಾ ಇದೆಯಾ?
Soil quality will improve, but also if we observe, the soil will be polluted because fertilizers will be made up of chemical substances which can harm organisms, which can increase fertility of soil.
Microorganisms, so chemical fertilizers ಹಾಕಿದಾಗ
They will it decreases. It cannot thrive. So it's not natural. Chemical substances will be more in fertilizers, so we can use manual and fertilizers. But fertilizers we must use in less.
what's the recommendation? Should we use more and more NPK chemical fertilizer or reduce it and do more natural farming?
Natural farm.
Thank you. Then IISC and Havan.
Good afternoon, one and all present here. I'm Leksha and this is my partner Sadia. Today we are going to be presenting about Madivana Lake's present state and its legacy. A little tranquil lake is more significant to our life than any big city in the world. Lakes. Why are lakes important to our lives? Lakes contribute a great value to human beings as they provide us with the life source which is water. Madivana Lake. Madivana Lake is one of the largest and the oldest lake in Bengaluru. It is believed to be 300 years old and was built by the Chola dynasty, the longest dynasty in South India. Madivana Lake is home to many plants and animal life. It ends by thus enhancing the biodiversity in urban areas. Like all the lakes, Madivana Lake is also a popular spot for bird watching, especially for migratory birds. It also serves as an important water reservoir and helps is 8 in the sample 1 and 9 in the sample 2. But the permissible length is from 6.5 to 8.5. The temperature is 26 degrees Celsius in the sample 1 and 28 degrees Celsius in the sample 2, and it continues so on. Our results were the high pH level, low dissolved oxygen, elevated turbidity, and moderate chloride levels. The nutrient level in this lake can increase due to dissolving of offerings like kumkum, haldi, and etc. Slight changes in pH due to human activity can disrupt the natural chemistry of the lake, making the environment unsuitable for sensitive species. These human activities not only increases the turbidity level but also makes it hard for the aquatic animals to live. We should avoid emerging toxic idols like which contains paint and other chemicals with heavy metals. We have to do proper disposal of offerings and avoid littering around the lake with plastic and non-biodegradable things which increases turbidity and harm aquatic life. We have to create awareness about the effects of pollution on lake ecosystems, emphasizing the need for the clean water and healthy environment for both humans and wildlife. Instead of emerging chemical idols, we can use mud to create the idols and emerge it which does not harm the lake. Conclusion. Festivals and other rituals are joyous occasions which strengthens people's faith and culture, which brings people together and make their culture healthy. These festivals should be celebrated, but these should not harm the environment. Instead, we can celebrate our festivals by honoring the nature. We referred these websites to create this PPT. We thank everyone for your support and for your cooperation. Thank you.
Immersion of adults is a very important reason for the pollution. What explain ಮಾಡ್ತೀರಾ?
Sir, in the idols, chemicals which are in the paint, so they can dissolve in the water and cause the water's pH level to increase. And also the heavy metals cause problem to aquatic animals, as aquatic animals are important. The things in idols which cannot be dissolved, they stay at the ground part of the lake, which increases the turbidity of the water. It's a problem for the lake.
ಬರೀ ಮೂರ್ತಿಗಳನ್ನ ನೀರಲ್ಲಿ ಮುಗಿಸ್ತಾರಾ? ಅದರ ಜೊತೆಗೆ ಏನೇನಾದ್ರು ಮಾಡ್ತಾರಾ?
Yes, sir. So they also put offerings like flowers and kumkum, haldi, etc.
Flowers ಹಾಕೋದ್ರಿಂದ ಏನಾಗುತ್ತೆ? How it affects the lake?
So it might stay there for long. So it is biodegradable, but it may harm the people nowadays use plastic flowers, which they dispose in the water, which does not easily get dissolved. So it also increases the turbidity of the water.
Actually, flowers ಹತ್ತಿದು ಎಲ್ಲವನ್ನೂ ಹಾಕಿರೋದ್ರ ಜೊತೆಗೆ ಪತ್ರೆ ಗಿತ್ರೆ ಏನೇನೇ ಪೂಜೆ ಮಾಡಿದಾಗ ಇವೆಲ್ಲ ಹೊಳೆಯೋದು ಶುರು. ಆರ್ಗ್ಯಾನಿಕ್ ಮ್ಯಾಟರ್ ಹೊಳೆಯೋದು ಶುರು. ಆ ಹೊಳೆಯೋದಕ್ಕೆ ಸಹಾಯ ಮಾಡ್ತಕ್ಕಂಥದ್ದು ಬ್ಯಾಕ್ಟೀರಿಯಾ. They use lot of oxygen which is dissolved in the water. ಅದರಿಂದ ಈ dissolved oxygen ಕಡಿಮೆ ಆಗುತ್ತೆ, ಡಿವೋಲ್ಡ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ. So aquatic animals they find lesser oxygen in the dissolved oxygen in the water and therefore they will die. So this is how immersion of hydals and along with flowers and other. So, what is the solution for this problem?
So for this we can use natural of mud and other.
Hydals, okay.
Sir, with guidance, we should not.
ನೀರಿಗೆ ಹಾಕ್ಲೇಬಾರ್ದು.
Yes.
ಅದನ್ನ ತಗೊಂಡು ಅದರಿಂದ ನಾವು ದೇವ್ರ ಮೇಲೆ ಭಕ್ತಿಗೆ ಸುಳ್ಳು ಭಕ್ತಿಗೆ ಹೂವು ಮನೆಯಲ್ಲಿ ಇದ್ದಿದ್ದು ಎಲ್ಲಾರು ತಗೊಂಡೋಗಿ ನೀರಿಗೆ ಹಾಕಿದ್ರೆ we are spoiling the water in the lake.
Yes.
So this is the lesson which we have to loudly share with the common people in that particular area, including our own also.
Yes.
Okay.
Thank you, sir. We depletes the oxygen levels, which is lessing the aquatic life is less over there.
Thank you. Well done. Vaibhav Karthik R and Purnesh R.
Morning everyone, my name is Purnesh. We are studying 7th standard of Algae Vilas Nirmangla. Today, for this big project, I have chosen Nirmangla Lake. Nirmangla Lake, situated near Nirmangla, Karnataka, is a very important lake for the surrounding water for the surrounding rural communities. This lake plays a crucial role in supporting local biodiversity and maintaining ecological balance of the region. Like many lakes in the region, it faces environmental issues such as pollution, encroachment, and inadequate maintenance. These issues have sparked local attention and conservation efforts. There are active initiatives by local citizens and environmental groups to protect and restore the water ecosystem and quality. The lake is surrounded by developing infrastructure, real estate, and notable landmarks, which emphasizes the need to preserve it amidst urban growth. This lake is surrounded. There is activities of human results in waterborne diseases such as typhoid, malaria, etc. Before 6 months, people have cleared 3 tons of garbage during summer and made it clear by monsoon season. And each citizen have spent 50 lakhs and 106 people have lost their lives while doing this job.
Main objectives of our project. Some of them are to investigate the human taken in the lake and to access the physical and chemical water.
Invertebrate lake location, perimeter 2.76 km, area 0.2 km square, latitude 13 degree 05 22.0 knot, longitude 77 degree 2447.4 east. This is the satellite video when in mobile and that is the aerial view.
Materials and methods. That water was collected from 2 different days. Species identification. We used Google Lens to identify the species. Instruction. These are the some tests that have have been undergone to test the water quality. Some of them are purity tests for ammonia, result oxygen.
Flora and fauna. These are a few photos of few photos of flora and fauna that we took near Binmangla Lake. Those are Clostridium, green algae, red worms, and Indian blue moth and Indian corn heron. These are a few snapshots which we took near while doing tests in our school.
That we got after testing as the ammonia and the nitrate quality are more with water. These are some things analysis. And the low dissolved oxygen point, poor water quality, ecosystem negatively. Steps like inputs, example from the sewage and agriculture runoff help.
Conclusion. Water should be treated before letting it to the lake. Construct a walking track around the lake to have access around the lake which help in monitoring and maintaining of the lake. Once in every 15 days, lake will be checked for floating waste. Just fishes should be introduced into lake water.
This is an online website which we took Binmangala Lake.
We would like to thank R&D department, our school and IISC to give us this full opportunity to present in front of all of you. Thank you.
Recommendation that introduce fishes into the lake ಅಂತ ಬರೆದಿದ್ದಾರೆ. Fishes ಇಲ್ವಾ ಈಗ ಬೆನ್ಮಂಗ್ಲ ಲೇಕಲ್ಲಿ? ತಿರ್ಗಾ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ರೆ ಅವು ಸಾಕು ಅಲ್ವಾ?
Sir, we can drink the water after the
First treat the water, then introduce the fishes. ಇಲ್ಲದೆ ಹೋದ್ರೆ ಈ ಕಡೆ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡಿದ್ರೆ ಆ ಕಡೆ ನೋಡಕ್ಕಾಗುತ್ತೆ. There is no use of introducing such a low quality water.
Any other any fish?
There are no fishes. Why birds come there?
Sir, it comes from the different migrating birds.
Thank you. Thank you. He, okay, okay. Oh.
Sir, yesterday when TVRAMACHANDRA sir was giving the he said that humans are including a few chemicals which are made the plants in aquatic have grown in tremendous speed. But as per to the oxygen level, if you compare it with the oxygen level. Sir, yesterday when TVRAMACHANDRA sir was giving lecture, he said that a few chemicals of human imports have made tremendous increase of aquatic plants in lake water. Sir, but as compared to the oxygen level, we compare it. It's good to know, sir, has it can increase the oxygen level.
ಈ ನೀರಿನ ಕ್ವಾಲಿಟಿ ಅಕಾರ್ಡ್ ಮಾಡೋದು ಮೈನ್ಲಿ ನೈಟ್ರೇಟ್ಸ್ ಅಂಡ್ ಫಾಸ್ಫೇಟ್ಸ್. ನೈಟ್ರೇಟ್ಸ್ ಮೈನ್ಲಿ ಈ ಮಿಷಿನಲ್ಲಿ ಕೆಮಿಕಲ್ ಫರ್ಟಿಲೈಸರ್. ಕೆಮಿಕಲ್ ಫರ್ಟಿಲೈಸರ್ಸ್ ಆರ್ ಮೈನ್ಲಿ ನೈಟ್ರೋಜನಸ್ ಫರ್ಟಿಲೈಸರ್. ಸೊ ಅಲ್ಲಿಂದ ವಾಶ್ಔಟ್ ಆಗಿ ಬಂದಂತ ನೀರೆಲ್ಲ ಕೆರೆಗೆ ಬಂದು ಡಿಟರ್ಜೆಂಟ್ ಎಲ್ಲರ ಮನೆಯೂ ಡಿಟರ್ಜೆಂಟ್ ಬಟ್ಟೆ ಒಗೆಯಲಿಕ್ಕೆ ಪಾತ್ರೆ ತೊಳೆಯಲಿಕ್ಕೆ ಎಲ್ಲ they contain high level of phosphates. ಹಾಗೆ ನೈಟ್ರೇಟ್ಸ್ ಫಾಸ್ಫೇಟ್ಸ್ ಜಾಸ್ತಿ ಆಯ್ತು ವಾಟರ್ ಲೆವೆಲ್ ಪ್ಲಾಂಟ್ಸ್ ಜಾಸ್ತಿ ಬೆಳೆಯೋದಕ್ಕೆ ಶುರುವಾಗುತ್ತೆ. ದೇ ಅಬ್ಸರ್ವ್ ಮೋಸ್ಟ್ ಆಫ್ ದಿ ಡಿಸಾಲ್ವ್ಡ್ ಆಕ್ಸಿಜನ್ ಇನ್ ದ ವಾಟರ್. ಸೊ ಬಿಒಡಿ ಜಾಸ್ತಿ ಆಗುತ್ತೆ. ಬಿಒಡಿ ಜಾಸ್ತಿ ಆದರೆ ಅಕ್ವಾಟಿಕ್ ಲೈಫ್ಗೆ ಡೇಂಜರ್. ಅಕ್ವಾಟಿಕ್ ಲೈಫ್ ಸತ್ತೋಯ್ತಂದ್ರೆ ನೀರು ಸತ್ತೋಗುತ್ತೆ. ಬೇರೆ ಬದುಕಿದೆ ಅನ್ನೋದಕ್ಕೆ ಮುಖ್ಯವಾದ ಇದೇನಂತಂದ್ರೆ ಅಕ್ವಾಟಿಕ್ ಲೈಫ್ ಚೆನ್ನಾಗಿದೆ.
Thank you, sir.
Prajna D. Prospect, Sri Priya, Krishnapur and IISC.
Pratiksha?
Namaste, Vanita. My name is Shasi. I am studying in 8th grade and I am from Purnapuram East. Today, my topic is all about diversity of trees in selected areas of Lal Bagh. Lal Bagh is a beautiful place to meditate, sit, work, research, exercise, and many more. Introduction. Lal Bagh Botanical Garden is dedicated for researching and preserving plant species while educating the public all around us about the tree diversity in my study area. This research has been conducted using the tree composition in my study area and the overall results have been that my study area is rich in tree diversity. Not only this, trees play a vital role in an environment. Trees contribute to the environment by providing oxygen, improving air quality, supporting wildlife, and many more. Objective, density and diversity of trees. In this project, I am going to assess about the tree diversity in my study area and share more information about the medicinal and other uses of the tree and most of the common diseases as a home remedy. Distribution trees and number of species of trees in my study area. There are data available on the trees of Lalbagh, but today I am here to present about the current state or current situation about my study area, how it is. This is the map of Lalbagh and these are the coordinates. This is mapping of my research area just to get the clear picture of my project. Materials and methods. Materials I have used in the study are A4 sheet paper for measuring the height of the tree, which is called tangent method, capturing photos, laptop for documentation, and measuring tape for measuring 20 by 20 meter grid. These are the results. Grid 1. In grid 1, the first tree is Indian beech. Its scientific name is Pongamia pinnata. Height is 14.5 meters. Girth is 0.9 meters. Its bark helps in curing hives and its fruit cough and cures diabetes. The third tree is woman's tongue. Its scientific name is Albizia lebec and it reduces cough and lung problems. Second grid. In the second grid, the first tree is sapota. Its scientific name is Mankara sapota. Its height is 11 meters and the girth is 0.84 meters. Its fruit helps us in controlling blood pressure, improves our vision and hair health. Fourth tree is Amla. The scientific name of Amla is Phyllanthus indica. Height is 12 meters and girth is 8.23 meters. Its fruit helps us in boosting our immunity power and improves our digestion. The fifth tree is white teak. Its scientific name is Melena arborea and height 22 meters, girth is 2.19 meters. Its bark is used for creating musical instruments. This is the third tree. The first tree is rain tree. Its scientific name is Samania saman and the other uses of rain tree are the seed of rain tree is used for making garlands. The third tree is peepal tree. Its scientific name is Ficus religiosa. It is 40.64 meters and the girth is 27 meters. Helps in digestion and it's very good for heart and helps asthma patients. Grid 4, the first tree is tamarind. Its scientific name is Tamarindus indica and helps in resolving respiratory issues and prevents cancer. Sixth grade, jackfruit. Its scientific name is Artocarpus heterophyllus. Height is 12.74 meters. Girth is 1.67 meters. Its fruit is anti and helps in healing wounds. Seventh grade. In seventh grade, the third tree is white silk cotton. Its cotton is used for surgical dressing. The major dependent white tree are honeybees. There are 25 to 30 honeycombs on my particular tree. Eighth grade. In the eighth grade, most of the trees are mango trees. The seventh one is champaka. The scientific name of champaka is Mongolia champaka, and it cures diarrhea and cough. These are the photos of my study area. These are the bar graphs which represent number of trees and species of trees in my study area. The first paragraph represents number of trees versus 8 grids. Similarly, the second paragraph species of trees in each grid. Lalbagh Botanical Garden showcases a remarkable diversity of tree species, where most of the trees hold a significant medicinal value in different parts, such as leaf, bark, stem, and roots. Mature trees are dominating the landscape than newly planted saplings. These trees are playing an ecological role by giving shelter and food to many other living organisms. Please do plant, care and support tree life. These are the references which I have referred to. I heartily thank TVR sir and Rujula Lana for this golden opportunity. This will be a beautiful experience throughout my life, and I hope you all enjoy my presentation. Thank you.
You mentioned so many
These are some dangers of our country.
study areas. How many of them are indigenous species and how many of them are exotic species?
I have read it. There are signboards, but most of the trees are from they're not native plants. Most of them.
Exotic.
Yeah. Most of them. There are trees from Australia, South America, Europe, and etc.
So indigenous species are a minority.
Yes, they wanted to showcase the species of trees.
What is your idea? Lal is maintained for such a long time. So now, as you said, is in a good position, in a good way. Other plants are coming good and healthy. So, do you think in every city they should maintain this? That's the best way to, control unnecessary things. Anything just to keep ourselves healthy is the better to do it as an albert in each country, in each city. Do you think so?
Gardens to plant in our surroundings also, not only in the botanical gardens. Like for one botanical garden is enough, and only there trees will be there. Only those trees is not correct. We have to care and plant trees in our surroundings also.
You go back to 16. Haripriya HR and Chinmay Moth.
My name is Chinmay.
Myself, Haripriya HR. We are 8th graders from Pune Primary School. Today, before we start our presentation, we would like to introduce a quote.
Without water, there is no life. Without blue, there is no green. So all terrestrial organisms struggle to survive without water bodies. These water bodies and lakes are recognized for and respected for their ecological and historical significance. These water bodies and lakes is the act of starts by storing large amounts of water. And apart from all these benefits, mankind has decided to pollute these lakes for the greed of money.
Our goal for this presentation is to observe the drastic change that can happen in water bodies in just a matter of time. And secondly, we will be comparing the water samples of Saraki and Madivana Lake. Lastly, we will document the different biodiversities of
The materials that we use are camera, phone for identification, book, pen, laptop for documentation, binoculars. The methods are observation of birds as a natural environment. And these are the total visits to the lakes.
These are our study areas, Saraki and Madivana Lake.
So, before 2013, Saraki was treated as a dump. Trucks would line up every day to throw our trash in Saraki Lake that created in a foul odor. And in 2013, a 10-day eviction led by a High Court order that started to clean the lake, costing the government almost 2,000 crores. Now, due to urbanization around the lake,
the lake is suffering severe problems and almost 10 lakh liters of sewage is being dumped into the lake. And recently, dead fish were recovered floating in the lake that tells that our cleanliness standards are still not being met. So here, this is a satellite view of the lake from almost 10 years. In 2019, you can see that the lake is almost dried up. That is because meaning the Bangalore Department Association wanted to rejuvenate the lake. That is because you can see here there are dark green spots indicating dirtiness. So the EDA completely dried up the lake, cleaned it, and then waited for the lake to flow. That almost costed 25 crores. So we tested the water quality of the lake and compared them from this year's March and 2024. And in the middle you can see this STN code. That means station code. The station code for this lake is 4504. So these are the test results, and this is algae. These are the birds, trees, and animals that we saw. So we saw these many birds. These are the scientific names, and these are the family they belong to. And one special thing that we love that we saw snake. We named it Quebec. You can see the photo.
Coming to Maniwala Lake, up until the 1990s, the water of this lake was completely potable, but now it is very polluted and it's not potable. And in October this month, dead snails and fish were found at the bank of this lake. And recently, before this month, an unconscious egret was found on the bank of this lake. And later it was found out that the egret drank the water of the lake, and it was the result of unconscious. And this is the satellite images of Madivada Lake from the past 10 years. And this is the test results of September and March. This is what we saw in the lake when we went there. We just captured all of the birds we saw and the plants. This is the table of the birds, scientific name and family. Same for the plants and trees. So when we visited the lakes, we saw that people and residents who are there, they used to dispose used items like pan masala packets and polythene bags. We also saw a bunch of dustbins which were fallen down. And wet dustbins were filled with dry waste. So we
came up with some of our own solutions. So like she said, we saw the dustbins, but most of the trash was on the ground. So we decided to make some signs and stick it on the dustbin that may create awareness in the people. And another way is to do it by scanning this QR code you see. That is, you can scan that to become something called a Lake Mitra. There you can contribute towards cleaning the lake. And the left one here is the QR code for WhatsApp, Sarki Lake's WhatsApp group. In there you can give ideas and plan timings to go and clean the lake. In the comparison between the two lakes, the table below shows the samples, pictures, and the purity of the water in front of the lake. From this information, our understanding is that Saraki is cleaner than Madivada, but however, Madivada lake is much richer in biodiversity than Saraki.
So based on our studies now, we have come to a conclusion that we cannot differentiate between these two lakes, as lakes can change, change through time, and they may get more cleaner or they may become more polluted. So due to the human activities around the lake. These are references for the project.
We thank all these people and
in these two lakes, which is more polluted?
Madivada.
So you cannot say that is better and this. You have to say this is more polluted, this is less polluted.
Yes, yes.
So both the lakes are polluted.
Yeah, it's polluted.
Only a difference of degree.
Someone, what to do?
in the QR code given over there, You can scan it on the WhatsApp group. So in that you can give ideas. To clean the lake, you can scan that QR code to become a timings and months, and you can even invite people to clean the lakes personally.
Work being done at the ground level to clean the lakes? Huh?
Sorry?
Any work is being done to clean the ground
Yes.
level?
Sarki is being
Giving ideas, QR code, that's one
Sarki Lake is being cleaned every month.
every month. This one, the other one, other one.
Madivana is actually clean, but yeah, the dumpsters are fallen, so the people over there are so they are rarely cleaned.
Fine.
Next one, Anantayam Rao.
And whoever presentation is, you have to come back because at the end we are certificates. Sir will issue. If you are not here, then you won't get the certificates. Back by 150. Listen, look, wait. Okay, at the end, if you haven't heard, be back by 150. Okay. You have to come back, otherwise you won't receive your participation certificate in the end. And you have to be back on time.
Students, next presentation is from Shripriya and Ragna. And please maintain silence. I can see so many people behind talking too much. Then I will make you sit separately if you continue to talk. Okay? Namaste everyone, I am Aditi. And I am Shripriya. And I am Ragna. Today we are presenting a project called Havan for Health. Now, I am going to introduce Havan. Havan is a scientific experiment in which special medicinal herbs are offered in a special fire pit. Havans have a direct effect on our ecosystem and it cures illness, mental and physical disorders. Havans can cure many types of neuropsychiatric diseases related to our brain like epilepsy, anxiety, depression and more. Havans have a number of benefits on mental and physical health. Objectives for this presentation are to get acknowledgement about the ingredients offered in the Havan, to know about the effect of Havan on the human body, and to learn whether the effect of Havan is good or bad for the environment. The materials we use for this presentation are listed here. Methods. We personally interviewed Purohits and Ayurvedic doctors and we did secondary research of online papers. The questions we asked Purohits are listed. Here, we have listed a few Havans and mentioned their ingredients. First Havan is Ganapati Havan. The ingredients offered in the Ganapati are known as Ashtadravya. They are listed here. Ganapati Havan is a Havan or a ritual performed to seek the blessings of Lord Ganesha, who brings victory and harmony in our lives. Next Havan is Navagraha Havan. The ingredients are listed. Navagraha Havan is a ceremony performed to honor the 9 planets that control every aspect of human life. The next one is Ayusha Havan. The ingredients are listed. Ayusha Havan is a ritual in which we worship the god of life who will bring us long life blessings. Coming to the fourth one. The fourth one is Mrityunjaya Havan. The ingredients offered in the Mrityunjaya result. Mrityunjaya Havan is a Havan or a ritual performed to honor Lord Sri Shiva and to overcome untimely death. Next Havan is Durga Havan. The ingredients are listed. Durga Havan is a ritual performed to seek divine blessings from goddess Durga. It is believed that she boosts self-confidence. Last but not least is Sudarshana Havan. The ingredients are listed. Sudarshana Havan is performed to seek protection from sudden death or dangers. Yajna resumes the brain cells, purifies the blood and prevents the growth of pathogenic bacteria which causes diseases. When sugar and ghee are burnt together, the smoke which is emitted can purify the environment and it kills the bacteria which causes measles, cowpox, smallpox and other things. The components when the supplemented vital element smoke is inhaled, it first reaches the brain, followed by the lungs and to the other organs. Thus, it heals brain-borne diseases. Clove is an important Havan Samagri. Clove essential oil, also known as CEO, has a significant effect in reducing the dose and duration of epilepsy. One of the main ingredients is when the smoke of ghee is inhaled, it clears the respiratory system and blood clots. Here, we have taken 2 experiments set up at Gorakhpur, 20 meters away from the level of sulfur dioxide and nitrous oxide before the Havan was 3.36 milligrams, 1.16 milligrams. During the Havan, it reduced to 2.82 milligrams and 1.14 milligrams. After
After the Havan, it decreased to 0.08 milligrams and 1.02 milligrams. The bacteria count in average water samples. Before the Havan, it was 4,500 milligrams. During the Havan, it reduced to 2,470 milligrams. After the Havan, it decreased to 1,250 milligrams. Now we can imagine how powerful these Havans are.
It is assumed that Havans are bad because anything when burnt emits carbon dioxide. This might be true, but in Havans, carbon dioxide plus water plus 112,000 calories gives out formaldehyde and oxygen. This formula shows that the carbon dioxide emitted in Havans plus the water vapor in the air and the 112,000 calories from the samagris is converted to a disinfectant and oxygen. Many volatile oils which have fragrance are emitted in the Havan. These oils will control the bad odors in the air. HCHO or formaldehyde is a very good disinfectant which purifies the environment. But this disinfectant only works in the presence of water vapor, which there is a lot of during Havans. Or samagris burn slow, which causes slow combustion, which makes this formula work. So, Havans contribute to curing the diseases and it has positive results. Each Havan has a specific purpose results and the ingredients would also differ from each other. Havans are performed with a lot of devotion towards the god, and I would like to say this and conclude that as per the opinion of Purohits, there are numerous benefits of Havans in which we have taken only a few and have did this, and there is a still lot more to discover. References we referred.
Now, I like to take this opportunity to thank all of my teachers who helped us and supported us. Then Shri Rujularana, Shri Ravi, they gave us IISC for giving us this wonderful opportunity for participating and Alvas for the accommodation. Thank you.
Very good, but as we suppose if very hungry, they started putting all the things into the oven, and you said, how can you manage? How can you, what can you say? And can you wait? And can you say, what is the answer? Asking this question, suppose in your family, if they are doing the, yoga, and they are putting lot of yes. So it is good to have. What do you say?
It's actually like interlinked. See, even Ayurveda is interlinked with all of this. So, even in Ayurveda, it has a lot of medicinal values. And in Havan also, it has many medicinal values. So, when it is offered, the smoke, it will satisfy us. Actually, all the, as she said the volatile oils that makes our mind relax and it makes us calm when it is inhaled. So that can relax us.
Good.
Thank you. Namaste everyone. My name is Anita. Today I am going to talk about my project, Comparative Study of Growth of Fungi with Chemical Fertilizer and Organic Fertilizers. In today's presentation, introduction, objective of the project, materials and methods, execution details, observations, results and discussion. Best way to introduce the objective of this project is to know what was and what is in terms of growing plants and crops. In earlier days, people used to grow plants and crops in an eco-friendly manner using organic fertilizers and organic pesticides. But in 1842, people started to grow plants and crops using chemical fertilizers. Why did this change happen? Because chemical fertilizers give us better yield in a shorter duration. But what is the harm in getting better yield in lesser time? To know the effects to our ecosystems. It has harmful effects on soil, water bodies, and all interconnected ecosystems. And as such, the objective of this project is usage of vermicompost of quality, quantity, and if it is a small area beside my house and started to grow fenugreek. Materials and methods. The materials that I used are chemical fertilizer that is NPK 90 and organic scale. Of 2 cross 1 feet, plots were tilled for sowing. And I planted equal quantity of fenugreek seeds to both plots. That is 1 tablespoon. I watered them daily and I added the fertilizers after the saplings came. The chemical fertilizer quantity that I put is 1 tablespoon mixed with 1 liter of water and 1 cup, that is 85 ml. Observe the growth for every 2 days and conclude which method is better. To gain I took knowledge about I took knowledge from interviews with experts. Yes, seeds were planted on 29th July. Seeds started sprouting on 31st July. And around 3rd August, many leaves started to emerge. And the addition of fertilizers I did on 7th August. Observation after the addition of fertilizers. On 11th August, the height of the plants in both plots were 2.5 inch. And in 14th August, the height in plot 3.5 inch and in plot 3 was 4 inch. On 16th August,
In plot 1 had an increase in the height, that is 4.2 inch, and in plot 3 was 4 inch. And so I decided to of the plants because the height of the plants were different. The average height on 22nd August came 5 inch in average, and in plot 3 was 6 inch in average. I added the second dose of fertilizers on 3rd September. On 7th September, the height in plot 1 was 8.26 inch average, and in plot 3 was 7.82 inch in average height. 3 was higher than plot 1, that is 9.4. These are some observations of leaves. On 9th August, the growth of the leaves in plot 1 spread everywhere, and in plot 3 was little spreading. On 14th August, leaves was narrow compared to plot 3. Continued till 9th September. And on 9th September is the leaves, color of the leaves. In plot 3 was light green, and in plot 3 was green. Did the harvest on 12th September. This harvest gave 111 grams, and in plot 3 gave 199 grams. Leaves on 12th September in plot 1 was looking dull, and in plot 3 was looking fresh. In plot 1, the color of the plot 3 was greener. Day on seed planting. This is 42 days after sowing the seeds. This is the average height of the plants as on 22nd August. From different angles. Average height of the plants as on 9th September. To summarize the results of the project, the average height of plants in chemical fertilizer fed plot was slightly short, and in plot 3 was slightly taller. Smaller, and in vermicompost had the color of the leaves in plot 1 looked compared to plot 3 compared to plot 1, plot 3's color of the leaves was looking greener. So the time of harvest of plot 1 was looking dull, and in vermicompost looked fresh. To conclude, based on the observation, we are seeing better quality of field in terms of height of the plants, size of the leaves, greener leaves, and fresher at the time of harvest. In terms of quantity, we are seeing higher yield in the higher yield of leaves from vermicompost. Check if vermicompost is a substitute chemical fertilizers. The soil quantity is to be measured after through soil testing. Samples are given for test, but results are awaited. I thank my school for giving me an opportunity to present, and I thank Fujilal and teachers to encourage me, and my Jyoti GM, my expert, and most important, I thank IISC for giving me an opportunity to present. I thank my parents for encouraging me. These are the references that I took. Thank you.
Your experiment is interesting, and it also confirms the activity of
natural farming, especially vermicompost, natural fertilizers over fertilizers, and experimented yourself. Tejas Kumar Sharma.
I updated and sent it. I just I have it right. Like I made some changes in the so can I give it to you right now? I have it. So I have the PPT. I can send it. I have the other PPT. I actually submitted it.
Yeah. Present this paper.
Namaste, Jai Shri to one and all present. I'm Abhinav Jain of BJ National Public School, and today I'll be presenting on the effects of boiling together on exotic species of butterflies. I chose this topic as I've been fond of butterflies since I was a child, and I've been mesmerized by their colors. So these are the contents of my PPTs, some objectives that I've set, the introduction, the methods that I've used to get the data, exotic and endangered species of butterflies and why they're ecology, modern day weather and how it affects these butterflies, and how do we sustain these butterflies, and then I will be concluding my presentation. Some objectives that I have set for my presentation are to study different species of butterflies and what role they play in the environment, to study the climate change and how they affect these butterflies, to assist in the preservation of butterflies, and to spread awareness about butterflies and resource plants, not only by neighborhood and my surroundings, but to the new members of the Bangalore Butterfly Club. Introduction. My project is about climate change and how it affects and saves species of butterflies. I researched different species of butterflies to find the role that they play in our environment. And I looked at climate change and how they affect these butterflies. My goal is to encourage biodiversity and protect butterflies and try to reduce the adverse effects of climate change on these sensitive species. Introduction to butterflies. Butterflies are insects belonging to the suborder of Parasita. They are usually characterized by their bright and colorful wings. They have a four-stage life cycle: the egg, the larva, which also has four stages: feeding, non-feeding, pre-pupal and pupal stages. Then comes the pupa, and finally the adult butterfly. These are the methods that I use. First is fieldwork. I've done nature walks, and I have identified a few species of butterfly. I've gone to Turchanapalya Reserve Forest, which the address latitude and longitude is given, and also to the cultivated forest ranges and arboretum to gain data. Then on the other, there is data collection and conservation planning, which is ongoing. I'm feeding the butterfly feed. During monsoon, most butterflies migrate to for suitable food, shelter, and breeding ground. So some commonly found butterflies migrating to Bangalore are the dark-blue tiger, common crow, and double-banded crow, and the common moth. Some observations are made that these butterfly species are found migrating back to their homes earlier this year. This might be due to the heavy rains experienced in the western ghats. This is caused by the southwest winds or southwest monsoons. This might result in some groups of butterflies taking advantage of a break in the rains to try to get back to their respective homes. But if the southwest winds or monsoons return before they reach, then it can disturb their migration and lead them to dying out. It could also affect the migration of the rest of the species of butterflies. Some data are collected. These are just three, but there are other species too. These are the butterflies you can see. Exotic and endangered species of butterflies. Some reasons for a few species of butterflies to be called exotic or endangered are they have low population, either because of climate change, habitat destruction, or human collectives. It could also be due to the inavailability of food because of them relying on fragile ecosystems. Some rare species of butterflies are the Kaiserian, Langus metal marks, blue morpho, and blue thunderbird. These are exotic, and a few of them are also endangered. The Kaiserian, also known as the Emperor of India, is found along the areas of 6,000 to 10,000 feet of elevation. For example, eastern Himalayas, West Bengal, Meghalaya, Assam, Sikkim, and Manipur. It has a strongest frostbite and flies at treetop range and in the presence of daylight. Some characteristics include its 190 to 120 millimeter wingspan and its habitat, which is the broad green evergreen forest, which is northwest. Next is the Langus metamorphs, which is a butterfly associated with Yankee dunes. But now, due to climate habitat destruction, it is now found in the Yankee dunes National Wildlife Reserve. These butterflies mainly depend on aegis and buckwheat as its host plant, and it is also the primary source of nectar for the adult butterfly. Some of its characteristics include 25 or 1 millimeter or 1.5 inch wingspan, and its habitat, as I mentioned before, are 80 centimeters. So this is some data that I made found, and find climate change. So these given here are some of the impacts of climate change on daily weather. So first thing is heat wave, which is a stretched period of heat, which could affect migration. Harsh and heavy rainfalls damage sensitive butterfly habitats. Droughts can also affect butterflies, but can also harm the butterflies in physical way. Melting glaciers and rising sea levels affect lead to shifting of weather patterns, which makes the old weather patterns unpredictable and leading to unusual weather occurrences. Urban events, which are cities having higher heat than rural areas, this could affect the butterflies' wings as they are very sensitive areas. Some of the impacts of butterflies are given. Habitat destruction, erratic changes in weather could cause heavy damage to sensitive butterfly habitats. Life cycle mismatches, higher temperature and upper weather condition could affect the timely migration and hatching of butterflies, which could lead to them not being able to find enough food and a suitable environment conditions. How can we sustain these butterflies? Butterflies are not only important for our visual satisfaction, but they are also important for our environment in many ways. For example, they indicate good environment and they also pollinate our gardens. The butterflies, but it is also a duty to protect their habitats. We can do these by the points given here. Conclusion. My study shows the results and efforts to boost biodiversity and protect butterflies. Through increased public awareness and planting more host plants, I have 10 to 12% increase in locally found species of butterflies. This positive trend underscores the effectiveness of conservation strategies and community engagement. Creating awareness and planting host plants has not only increased the number of butterflies, but also led to the rediscovery of butterfly that was not seen in Bangalore and the rediscovery of species that was sighted. For example, the yam fly, which had not been sighted in Bangalore, which is a rare site, now is sighted in the Comparative forest ranges for about a year. And the conjunctive fly, which was observed 13 years back, has now been rediscovered and is undisturbed. My findings highlight the role of ongoing conservation and maintaining ecological balance. I would like to take a pledge because I pledge to do my utmost to protect both the habitats, butterflies, and butterflies. But I would like to accept my legal gratitude first to the judges, knowledge is very appreciated. Secondly, to my teachers, for without them I wouldn't be here. And I would like to thank my parents for their support and being here with us. Thank you.
Tejas Kumar. Sharma.
Morning, Jayashree Gurudev. Myself, Tejas Kumar Sharma. I'm from Bengaluru, and I'm going to present my study of Dragonfly Lake. It's like a comparative study between the years 2022 and 2020. So, first of all, about Dragonfly Lake. Dragonfly Lake is located in Bengal. It has a perimeter of 3.05 kilometers and has an area of 137 acres. And this is the water and soil collection. I've collected the samples on 4th August during evening 6:50 p.m. and this is the exact location where I'm from. So these are the contents of my project: objective, water test, soil test, and microorganisms. My objective in the study is the chemical, physical, and biological parameters of Dragonfly and compare the data of 2022 from 2024. And my aim is to suggest ways to improve the situations of Dragonfly. So these are the water tests: pH level, calcium, phosphate, carbonate, magnesium, chloride, and sulfate. These are the water tests. First of all, pH is 8, which shows that the water is alkaline, which is not very good for the lake water. Like too much alkaline water is not very good for supporting life. First is the carbonate test, where I have added sodium bicarbonate with the lake water in a test tube. The result should give a bubble brisk effect, which shows that the carbonate is present. But I have not found that in the soil water, which shows that carbonate is absent. Next is the sulfate test, where I added barium chloride to the lake water, which should give it a white color or white precipitate, which was seen in the result. So I can also conclude that sulfate is present in the water. Next is phosphate, where I added ammonium molybdate and nitric acid to the water, which should give it, diluted nitric acid with ammonium molybdate, which should give it a yellow precipitate or color. So I have not seen that in the result, so I can conclude that phosphate is absent in the lake water. Next is chloride, where I added So in the lake water, I added silver nitrate with the water, which should give it a white color, which was seen in the result. So I can conclude that the chloride is present in the water. Next is calcium, where I added ammonium oxalate and the lake water, and in a test tube, the result should also give it a white color. The result was also seen in the test tube, so I can also conclude that calcium is present in the water, in the lake water. The next one is magnesium, where I added ammonium chloride, ammonium hydroxide, and sodium ortho hydrogen phosphate, which should give it a white color or white precipitate. The result came as in the same way, that there was white color formed in the test tube. So I can also conclude that there is magnesium present in the water. That's it for the water test. This is the main table for it. pH is 8, sulfate present, calcium present, chloride present, phosphate absent, magnesium present, and carbonate absent. The next is the soil test, under which I'll be doing pH level, moisture content, and the soil NPK test, nitrogen and phosphorus test. Under nitrogen test, there will be two more, nitrate and ammonium. And in nitrate, there are two types of tests: brown ring test and copper chip test. I'll start with the brown ring test. I'll start with the pH and moisture. The pH is 8, which is also alkaline, same as the lake water, which is not very good for life conditions, but it's still okay. And the the moisture content, there is, I took 78.889 grams of soil. After heating it, I see 2.37 gram of decrease. So we can say there was a good amount of moisture in the soil. Next is the brown ring test, where I added iron 2 sulfate and concentrated sulfuric acid. The result should form a brown ring between two liquids, which shows that nitrate is present in the ring, which was not seen. So, and in 2022 it was shown, but in the current situation 2024 it was not seen. Next is the copper chip test, where I added sulfuric acid and copper chips. I heat the solution, which fumes, which was not seen. That means nitrate is not there through copper chip test and the brown ring test. But in 2022 it was present, and in 2024 it is absent. So we can confirm that nitrate is absent in the soil water in the current year. Next is the ammonium test, where I took diluted sodium hydroxide and I heated it. It should give an ammonical smell, which shows the presence of ammonium. I added, it's written fumes. It should give an ammonical smell. In the 2022, this ammonium was present, and in 2024 it was absent. Next is the phosphate test, where I added concentrated nitric acid and ammonium molybdate to soil water, and the result should be the solution should give it a yellow precipitate or color, which was not seen in 2024, but in 2022 it was seen. So ammonium and nitrate both are absent in 2024. Yeah, that is the table. In 2022, nitrate and ammonium both are present, and phosphorus is present. And 2024, both of them are absent. And the moisture content difference is 2.37 grams, and pH level is 8. And the last one is the microorganisms, where I took a test tube of both soil and water, lake water, and I took the soil water, and I centrifuged it to see it in the under the microscope. In the soil water, there were no moving organisms, but in lake water, there is one type of bacillus bacteria, and I saw another unknown macroorganism in the lake water. And these are the references I have used, and I am going to give you the recommendations for the lake. We should stop the rapid construction around the lake, and we should also avoid construction around the lake. And due to urbanization, we have seen many problems in the lake, and it is not good for life to support it. And the last thing is, we should take proper measures of maintaining, and time to time, periodically we have to test the water and soil to see the conditions of the thing. Thank you. Further questions? These are the other pictures.
2022 ನಲ್ಲಿ phosphorus present, 2024 phosphorus is absent. Hello? Yes? No? 2022 the lake content phosphorus was present, in 2024 it is absent.
Yes, it's absent.
What happened?
By doing the qualitative test, I didn't get it. Yes, I didn't get it. Like, it's not there in the PPT. I can't show the new PPT which I made, but phosphorus was absent in the.
No, it's absent.
Yeah.
What's the reason? And
Reason-
what's the implication?
The reason, because of the urbanization, or maybe like new nutrients like going away because of the formation of like other stuff, and because of the disposal of waste in the lake. That might be the reason. Thank you.
Srivatsa, please. Yukta Vivek Tatke. Yukta Vivek.
My name is Ratna. I'm going to talk about conservation and restoration status of lakes in and around Pune. These are the things I'm going to talk about here. My objective was to get conclusions and possible solutions by lakes. Most of the data I've taken is from BBMP and NGO sites. With the lakes first hand, comparative with earlier data from 2018, and most of the pictures in the same station will be taken by me. The materials which I used were cell phones, conservation study. Sadly, only about 210 lakes remain in the land of 1,000 lakes. The need for conserving and protecting our lakes is now more than ever. Lakes improve the quality of life in many ways. When we compare an old 1878 map of Bangalore with today, we can see that the orange circles indicate the lakes which are conserved and the red circles indicate the ones vanished. In this region, we can see more or less all of the lakes are conserved. We move on to restoration. I had started with Puriwan Lake first since it was closest to our lake and had earlier data. But then it's currently dry, so we went to Vartigar and Basavanapura lakes, which are upstream Puriwan. We also studied Kalitamla and chose Golahali for non-BPMP lake. To check restoration, we studied infrastructure, wildlife, bird life issues, and water quality. In this table, you can see that Kurikuma Lake has the least infrastructure. Kotigere is a Basavanapuram and Kalinakara are way better. Lake's fencing is region places. There's a temple in the lake which attracts a lot of garbage. This lake just justifies the reason why the coordinates and data are different on BDMP documents and Kotigere Lake is partly fenced. The installation is in process and it's getting better, and the triangles are in place. Basavanapuram is a fenced lake and has strict timings. Triangles are in place and it's a restored lake. Kalinakara lakes in this set, and it's the only lake which has a net at the inlet to avoid debris coming in. The earlier was restored in 2022, but it's very dirty. Now it is hardly maintained. And there's also a KV substation nearby, which was requested relocated and it's still there. We move over to the bird life. In July, the bird life of Holima was mostly concentrated in this shallow pool, but then right now that is covered with water hyacinths. That wildlife is gone. Kotigere Lake's bird life was mostly in 50 days ago recently, they filled up the lake and most of the bird life is concentrated at the island and wetlands. And as the lake is full of water, even ducks may come through. Basavanapuram has a limited bird life and limited ecosystem compared to Holima. We also saw a kingfisher which tells us that food chain exists over there. Comparing bird life of Holima Lake 2024 versus 2018, there were a lot of birds in 2018 in numbers as well as in species because the lake was full of water. The most interesting one was the pelican, as it told us that the vast full ecosystem was present. Let's look at the issues the lakes have. The outlet of Holima Lake is carpeted by thick water hyacinth. The Raj carpet too is not maintained, as there's a lot of sewage flowing and garbage thrown in it today. The situation at the inlet is increasing. Like it's getting better since TVMT is doing some construction work over there. It has shrunk over the years. In this slide, you can see that the lake is deprived of water mostly for conservation activities. Restoration activities, sorry. Kotigere Lake's restoration is in process. The entire lake was covered with water carriage. Since BBMP finished the work at the inlet, they filled the lake with water. We don't know if they took the water carriage out before filling it with water. Basavanapuram doesn't have a lot of change, except the beach has grown a lot. The water level is still the same. Kalinakara also doesn't have a lot of changes. The color of the water is still clean. The water level is also same. But one good thing is that there's a full family of cormorants living over there right now. The situation has gone from bad to worse. You can hardly see the lake because the beach has grown a lot over there. The conclusion which we reached so far are Basavanapuram and Kalinakara lakes have been restored, but then they don't have a full ecosystem yet. Kotigere Lake needs some upkeep, and Holima Lake is showing a lot of promise as it is being restored. We look at water quality now. This data is taken from KSPCB's water quality data. They publish it for approximately 110 lakes every month. They publish it for some 33 or 37. Based on CPCB's classification for water usage, here I want these 10 parameters. We study these for 3 lakes since Holima Lake's data is not available. This slide is just an example of how the data comes. In this table, as you can see, it is color coded and each color is given to one class of water usage. In this table, as you can see, dissolved oxygen, fecal coliform and total coliform have variations while other parameters are stable. BOD, total coliform and dissolved oxygen are the main challenges faced, and most of the lakes are class D. Historical water data of Holima, the dissolved oxygen changes from inlet to outlet. The BOD is high in both inlet and outlet. The pH is very alkaline, but it's still very roughly. The possibilities for improving water quality data. Water quality for Holima, for Kotigere is important as it is upstream Holima. We can also put floating wetlands and planting aquatic plants could be another method to improve. If we do things right, we can give nature another chance. We look at Chernobyl for this. last time the world scientists said about 20,000 years of the radioactivity ago. They only now started to see quite some wildlife for there. This is a very refreshing lesson for us as we just learn. We give nature a chance and yes, it will do its bit. The current installation of water quality data showed us this. We should also consult with experts and should learn from other installation activities. If we do things right, we can get Fulimo and the other lakes that you back. Life always finds a way, you just need to give it a chance. IISC and Algae College for giving us this opportunity, and I would also like to thank my teachers for guiding me through this project. These are my references. Thank you.
So, two contradictory pictures. On the one hand, you say major portion of the lake is covered with water hyacinth and other plants, and the water quality is also good. On the other hand, another picture shows lot of birds coming there and foraging. How do you reconcile this contradiction? Lake quality is not good. Lot of lake area is covered with water. How come so many birds come there for foraging? What do they eat? Why do they come there?
Eagles will come to eat insects over there, which go on the water cabbage. The water cabbage, I think, was up for verticated. So the water cabbage in verticated was. That's why, like, the water cabbage was. 15 months ago, they changed it. Being fed with water. In a month or two, even ducks and the other wasn't used to take out.
I suspect another. It showed Chernobyl area there. Animals have started living. I suspect our birds have changed their food structure in such a way that they have started eating anything that is available in the dirty lake. Their food habit has changed and degraded also, so that they can survive in the degraded lake also.
Yeah, they have adapted to the lakes being polluted, but then it's not good because even the birds are falling sick. And in Chernobyl, the animals they have like their immune system has changed and they've gotten used to the radioactivity and that.
All the students who have presented papers on different lakes of Bangalore, we would like to study this aspect also. Whether the food habit of the birds have changed in such a way that they can survive in the dirty lakes also, eating whatever is available. Okay. Thank you. One minute.
Showed us history is going on nowadays in every way. I mean, not only in your own opinion, how you can help to
I joined cleaning activities and after planning to like write an email to PPMP about all these problems just in case they don't know, and I will make sure that it's completed. Thank you.
Arjun. Prashant, Manish, and Sri, Sarvesh.
I will be presenting on the role of artificial floating islands in mitigating freshwater algal. So now let's talk about the problem, algal blooms. Algal blooms refers to the rapid growth of algae, which are tiny green plant-like organisms that accumulate on the surface of water bodies. Excessive algal growth has various negative effects, such as reducing the clarity of the water, inhibiting the plant growth, and it can also lead to fish kills. Ultimately, all of this will lead to an algal bloom where no organism can survive. Let's talk about the current remediation strategies. Then ultrasonication, which is the use of high frequency sound waves to drown the algae. Mixing, which is the physical mixing of water to avoid stratification of nutrients. Aeration, which is to increase the oxygen levels of the water. There are also methods such as the use of algicides and biological methods extracts microorganisms. Challenges is that they may be less efficient, higher in cost. They may need reoccurring treatments, which means that they're not long lasting. Some of these may even be hazards to the environment. Our proposed strategy is to use artificial floating islands as a way to reduce the algal blooms. On the artificial floating island, we have used the plants Colocasia and Vetiver. Our objective of we demonstrate that artificial floating islands control algal growth by lowering the nitrate levels in the water. Our second objective is to assess the capability of Colocasia and Vetiver, which are used in air. So the question arises, what is an AFI and why an AFI? An artificial floating island is a human-made platform designed to float on the surface of water bodies. They support vegetation and have various ecological benefits. AFIs reduce nutrients like nitrogen and phosphates in the water. They regulate the amount of sunlight that enters the water. They increase the biodiversity by allowing birds to lay their nests on them and by tadpoles to take shelter in their roots. They are immune to fluctuating waters and they can be implemented in water bodies. Let's talk about the properties of Colocasia. Colocasia is also known as tallow or elephant ear due to its large heart-shaped leaves. Its Colocasia esculenta. Colocasia is a perennial plant that thrives in tropical and subtropical regions. It prefers a wet, marshy environment. The roots of the Colocasia plant are fibrous and extensive. This leads to nutrient uptake. It also has a thick fleshy stem called the corm. This stores the nutrients. Colocasia has broad leaves which maximizes the photosynthesis. Colocasia's continuous growth ensures the continuous nitric absorption. This makes it a good, reliable, long-term solution on AFIs. Now let's talk about the properties of vetiver. Vetiver is also known as lavanja or vetiver. It's a perennial bunch grass of the family Poaceae. Its botanical name is Chrysopogon zizanoides. Unlike other grasses whose roots grow vertically, vetiver's roots grow vertically. This makes it more stable on the neophytes. Vetiver is non-invasive, which means it doesn't spread uncontrollably. Vetiver requires low maintenance and is tolerant of extreme weather conditions. It's known for its ability to filter pollutants in the water, such as heavy metals, nitrates, and phosphates. This is our experimental setup. We took a tub and filled it with 20 liters of tap water. We figured out that the initial nitrate level was 20 ppm of nitrates at the starting of the experiment. We maintained control, which was water without the plants. We placed 5 colocasia and vetiver plants in floating grid. We maintained similar conditions for both the experiments. Actually available nitrate testing kit. This works by adding a reagent to the water sample. It reacts with the nitrates and produces a color change. The color change is compared to a color chart which is provided. It provides a semi-quantitative measure of the nitrate concentration. The results. The initial nitrate level was 20 ppm of the at the starting of the experiment. The final nitrate level was reduced to a range of 0 to 5 ppm. So that we maintain consistently show 20 ppm of nitrates throughout. As you can see, the has performed slightly better than vetiver in and similar results were shown for vetiver as well. Lakes in Bangalore that have implemented AFIs. Kannamangala Lake, Alsur Lake, Agara Lake, Madivada and Kuntenkadi Lake. You can see that the AFIs have helped in the restoration and health of these lakes have shift our focus to Kuntenkadi Lake. This is our study area of Kuntenkadi Lake. It has a perimeter of 920 and its area is 55,500 and this QR code will take you to YouTube where you can see more on the Kuntenkadi Lake. And evolution of Kuntenkadi. In the year 2009, the BBMP started the rejuvenation of the lake. In 2013, the lake got covered with weeds and the rain due to this weed in the sky now. In 2016, AFIs were introduced into Kuntenkadi Lake and testing and analysis of the water sample was conducted. In 2018, sewage from an adjacent layout led to a massive fish kill. In 2019, an underground drain got connected into the lake, in all the work. In 2021, deweeding was done and the fountain was installed. BBMP deflated the lake in this 7 AFIs and 2024, which is present, the lake is in good condition. These are the nitrate levels of Kuntenkadi Lake. The nitrate levels have considerably fallen from the year 2016 to 2024. This shows that the water quality has improved in Kuntenkadi Lake. The additional conservation efforts done at Kuntenkadi Lake. So the PVC pipes which are used to make AFIs are replaced with bamboo ladders. AFIs are cleaned regularly and a fountain has been installed to aerate the water and address low oxygen levels. Dry leaves are used along with dry leaves are used along with cow dung to make manure, and various trees have been planted around the lake area. This forms a green canopy around the vetiver plants in colocasia plants and vetiver grass are effective in reducing nitrates in water. The effect of colocasia and vetiver may have other effects on such as pH, turbidity, dissolved oxygen, heavy metal content. And we should also explore other combinatory methods such as this is a picture gallery. Acknowledgements. We extend a heartfelt thanks to our school, ACS Magnolia Bharati Public School, for their great support. We want to express our gratitude for Dr. Vinay Kumar, Mrs. Usha Rajgopalan, and Mrs. Divya Shetty and Mr. Nataraj Pandit for their invaluable expertise for their project. We would also like to thank the IISC for providing us with here are the references. Thank you.
AFI method.
Yes, sir.
AFI method appears to me like this. It's a temporary method of making a dying lake appear living. it does not permanently solve the problem of dying plants. What do you say?
Sir, AFIs are not a solution to algal blooms, but they maintain the nitrate levels. And this is what I wanted to say. So AFIs, if you place AFIs in a lake that's covered in algal blooms, it may not prove to be so effective. However, if you place AFIs in a healthy lake, and AFIs are shown to be effective in this type.
Lake where the water quality is very low, AFIs will not solve the problem.
What I wanted to say is AFIs cannot, they themselves cannot kill the algal blooms, but they maintain the conditions of the water quality. They improve the nitrate, they decrease the nitrate. Thank you, sir.
Ardra and Rajeshri.
Everyone, myself Ardra TS. Myself Rajashri HK. We are representing Macroinvertebrates Girls High School. We are from 8th standard. And our project is about waste management, Samrani ash as fertilizer. Action. A project is about how to minimize any kind of waste generated at home or how to effectively plan waste generated. And it is believed that the smoke released by burning the samrani has some soothing effects. And as we all know that incense sticks are and in most of the houses along with incense sticks, cups and cones are also lit for fragrance. But however, we rarely think about the ash we carelessly throw away along with wet waste or dry waste. Objectives are to study the impact of ash of tooth cones or cups if disposed with garbage, understanding the chemical nature of samrani ash, analyzing the possibilities of using the solution of ash as a fertilizer for plants, creating awareness about managing waste at home itself, and contributing to the environment in our capacity. Materials which we use for our study are dhoop and incense stick ash, weighing machine, water bottles, pots, garden soil, fenugreek seeds, logbook, and a pen. Why are dhoops used? Incense sticks and dhoops are an integral part of prayer in many families, and the aromatic smoke released by burning the also proven that the volatile organic in the incense interact with the organic responses in the body. Picture of the Asian styrax. In ash are carbon, potassium, magnesium, and phosphorus. So these are considered as plant nutrients. So we can recommend the usage of samrani ash at least for ornamental plants. So usually most of the households dispose the samrani ash along with other garbage. But when animals ingest this waste, this ash along with the vegetable or fruit waste, they can get gastrointestinal irritation as well as vomiting. Weight calculation of the ash. First, I wanted to know how much ash is generated in micros. So our project was from July to September, and of Samrani ash in water. We added water to a small portion of ash taken in a beaker, and the resultant liquid appeared to be a solution. Test as well as we used indicators such as methyl origin phenolphthalein. So when we did litmus test, we got to know that the liquid is a base as litmus showed a color change. And when we tested with indicators, we again got to know that the liquid is. Later we prepared few pots for our study, and we collected garden soil in the pots and marked the pots as A, B, C and D. And we sprinkled dope ash solution in pot A. We mixed the soil of pot B with dope ash, and only water and soil were used for pot C and pot D. Sowed fenugreek seeds, and the day of sowing was 4 July, and we observed the sprouts after 4 days. Pot A and B had sprouts, and C and D showed no sprouts. The dope ash solution in pot A was sprinkled once in 3 days, and the remaining days we sprinkled only water. After 6 days of sowing was that most sprouts were in pot A and pot B, and no sprouts in C, and 2 sprouts were in D. So next we repeated the experiment with the second sets of pots, and the soil and seeds selected were of the same. So our observation was that pot A showed sprout in 3 days. Pots. This time we kept the pots on the terrace to avoid the rodent attack. Our observation was that as in the first and second experiment, the sprouts started appearing in pot A itself, and the growth rate in these pots were also known. Final set of pots. So experiments we noticed that the sprouts started appearing in pot A itself. This was the fourth set of pots. As you can see, more sprouts in pot A. And this was a picture taken when we were asking our schoolmates to collect the Samrani ash from their house and to clean it so that we could collect it. Of estimated generation of dope or agarbatti ash per house. We asked each volunteer to collect the dope ash from his or her house in a box and Monday to Sunday, 7 days, and to bring it on the next Monday. And we kept a box in the school to collect it and monitored how many members brought the ash. And we weighed the ash collected and divided by the number of students to find the average. We got an average of 3.2 grams per house, and we had 5 houses and 16 grams was the total weight. The number of houses were 5 and the average production was 3.2 grams, and method of dope ash disposal was waste or dry waste, and the type of incense used was cups, cones and sticks. And average dope ash generated in a week, we got it as 16 grams, that means 2.29 grams per day. And average dope ash in a year, we got it as 835.85 grams. So if we calculate all the houses and temples, the amount of ash generated will reach hundreds of kilograms. So our study is aiming at converting this ash into something useful.
Points to compare the growth of fertilizer organic manure are used. So we took normal soil, fertilizer soil, soil with solution was added, and soil with organic manure. We saw that in organic manure, fertilizer and normal soil, we saw the growth of saplings, and 2 pots with Samrani ash solution showed growth of more saplings despite the rain flooding in the pots. We conclude that in all the 4 cases of our study, we found that the seeds in pot A with Samrani ash solution germinated faster compared to other pots and other saplings, and it proved that the ash could be used as a fertilizer. And the study also proved that using the dope ash solution in water is better compared to adding ash directly to the soil. And since a detailed study is required about the chemical contents in these ashes, for now we only recommend the usage of Samrani ash for ornamental plants. Are that overdumping of ash can have a negative impact on the environment and human health,
as insensitive ash contains toxic heavy metals including copper, chromium, nickel, cobalt, etc. And so the disposal of this insensitive ash in rivers and other water bodies can threaten the living organisms due to the presence of heavy metal and high alkali content. So it is better to use it as a fertilizer for garden plants. And an account is created need to be generated in the public itself. And the manufacturers of the puja goods will also be made aware about the future utilization of these ashes so that they'll be careful with the ingredients. Acknowledgement. We sincerely thank CS Department and IISC Bengaluru and Dr. TVRAMACHANDRA and his entire team for giving us an opportunity in Lake Symposium 2024. We also thank our school management, teachers and schoolmates for providing us with all the support and cooperation. We also express our gratitude to our school security and helpers for in our project, especially for taking photographs. These are some references.
Experiment is very interesting. Stories of really they have showed that it can be used as a fertilizer through a sincere experimental method that sincerely appreciate your experimentation, attitude, and the
No one has to get worried about commercially. It's a good thing.
We don't have confirmation that these incense sticks don't have chemicals, right? Harmful chemicals.
As a they may be preservatives like it can be stored for so it may be harmful if we use it for other plant and ornamental plants.
Like the few plants might have some good type of chemicals in it and few might not. So we need to classify it, right?
But almost all the ashes they are rich in plant nutrients and they can also have chemicals. There are ashes without chemicals sometimes.
How do you have confirmation that these one it is recommended them for gardens and you usually grow herbs so
Not like flowering plants or they can
like once you use it it's like in the soil, right? The soil will get affected. You can't
it will get affected. That's why we had mentioned in our recommendations that the manufacturer should also be careful with the ingredients.
Adhyarao and mitigation. Adhyarao.
Afternoon everyone, myself Adya.
Myself Hitaishini.
We are studying in 8th grade.
And representing Bangalore Place School.
Our study is on native plants. Introduction. Nowadays we find that there is an interaction between the climatic change and the degradation of the ecosystem. Native plants are adapted to the local environment and are vital habitat for birds and butterflies. It has been reported that many birds and butterfly species which were noted in our area once upon a time have been vanished or their population has been minimized. Our study is based on secondary data. We also attempted to grow these plants by sowing the seeds and distribution of seed balls. Objective: to study the contribution of native plants to an ecosystem, to understand the native plants, to analyze the way to revive the population of native plants, to study the rate of seed ball in revival process. Materials used: seeds of malva almond and jamun tree, sieve, garden soil, water, gloves, logbook and pen, trays. As we celebrate malva neighborhood. Yes, fruiting season July to September, germination 6 to 8 weeks, fruiting period 2 to 3 years, ecological importance, drought resistant, suitable for reforestation and also provides mulch to the soil. Medical advantages: fruits and bark used for treating dysentery and leaves and fruits for astringent. Types of soil and climate: well aerated, moist and sandy soil. Ecological significance of malva almond. The tree near our area is quite big and also provides shade to the pedestrian. Since it is drought resistant, it is an ideal choice for reforestation. The tree wood is used for making furniture and cabinet. The tree's bark, leaf and roots are used for tanning, leather preparation and bed spoiling. Its roots grow horizontally over a large area and thus can prevent the soil erosion. It is also said by fish breeders. They use these leaves in their aquarium to lower the pH level and also heavy metal content in water. Malva almond is host to the following butterflies: large oak blue, red flash, red spot, dingy line blue. The grey-bellied trees will also reported to feed on ripe fruits and seeds of Malva almond. Several fungi and birds and bats are also used to feed on fleshy fruits and drop the seed everywhere. The causes of disappearance of Malva almond, climatic change, habitat loss, and pests are few reasons for in population. Longer duration of growth. It nearly takes about 20 to 25 years to grow. This might be the reason why it's not much widely preferred as an option.
Lifespan is more than 100 plus. Fruiting season is May or June. Germination period takes about 10 to 15 days. Fruiting period is 8 to 10 years. Ecological importance is provide shades and habitat for animals, birds. It's a source of iron and ascorbic acid. Dried and powder seeds of jamun seeds. Diabetes. Type of soil and climate. Deep loam and well-drained soil. Some of the which feed on jamun fruit, green pigeon, parakeets, Indian greyhound. Jamun tree is also larval host for large oak butterfly. Causes of reduction: we learned that these plants were very common in our area, but nowadays most of the time we see these following plants which are planted in Bangalore: trumpet tree, jacaranda, contrast lancifolius, an exotic flowering plant is planted by BPMP in Yalanga. This article was reported in Deccan Herald newspaper 10th May 2024. These are the pictures of the invasive species which were kept ready for the distribution. Seedball preparation, distributing of seed balls. Soil space in our school are managed, right? So we place the seed balls in the empty site. Observation after 15 days. The seed balls did not show any sign of germination. Some of the seeds got crushed under the vehicles which were parked. The seed left in the pavement tools. Preparing jamun saplings. Since we did not get a positive to the manual method, we took 10 pots and filled them with garden soil and placed hurdles we faced. Initially, we kept the pots in the cellar. Destroyed them. We shifted the pots to the terrace. Due to occasionally rain, the saplings were spoiled. The seed was where we placed in the empty site. After a few days, weeds like party, and so we could not observe the germination. Classmates were doing soil study and they said, days when they took soil for study, they told they found guava trees more. This might be because common might dispersal of seeds. We found this true because we have two. We thought of extending our studies with other native species to check their
List of plants included in BBMP's supplemental planting model. In dry deciduous forest, 14 types of plant species were proposed. Moist deciduous forest, 17 types of plant species were proposed. Semi-evergreen and evergreen forest, 19 types of plant species were proposed, but none of these included Malva almond or geometry. But most of them were honge, acacia, sitafal, etc. This data is officially issued from Office of Deputy Conservator of Forest, BBMP, dated 10th August 2023. Some of the anomalies found in the proposal: Custard apple, which need regular watering, is included in the plantation of dry deciduous. Custard apple require 120 to 140 centimeter of rainfall, but in dry deciduous, the rainfall is 70 to 100 centimeter. Acacia ariciformis agave, silver oak is not native to India, but is included in the proposal. Silver oak is less effective at storing carbon than other native plants and also can be invasive in nature. In fact, jamun tree stands one of the best choices after neem trees for plantation. But surprisingly, these fruiting trees are not included in the proposal.
Native plants help to maintain sustainability when native plants are replaced by exotic varieties, the animals, birds, insects, butterflies related on them will be affected and their population will be minimized. In some extreme cases, exotic species can become invasive. Native plants can be difficult, hence prevention is better than cure. Recommendation trees must be given priority by the forest department while giving compensating plantation programs. BBMP, the Koti Visha Saniya program with residency, welfare associations and school students must include only native species of plants. See, we can distribute these seed balls in
Thank TVRAMACHANDRA sir and his whole team for giving us an opportunity to present our project. We also thank our school principal, teacher and student for guiding us and cooperating with us. We also thank our senior who helped in preparation of seed ball. We also thank our school secretary and us because they helped us in necessary item required and also our works. These are some of the references. Thank you.
I really appreciate your advocacy for the native species to be planted by the BBMP or any other government bodies or even social organizations. First priority must be given for local species. And exotic species may be grown in areas such as Lalbagh, which are mainly for ornamental purpose. And therefore, I highly appreciate your paper advocating for top priority being given for local species such as jamun and albar, almond, and such other species. Thank you.
Is there any specific reason for choosing only these two plants? In our areas, jamun and almond tree. Is it a native plant? Yes.
Okay, my question is that you said the almond tree takes about 20 to 25 years to grow, so it's not that popular as what you said, right? Is there any, there are methods which makes it grow faster. So can you do that? It has so many advancements which can be used and it's very beneficial for humans.
But it to age for fruiting and all. So manual method.
Okay, fine. You do that after a certain period of time. If you take the first few years until it gets mature, you can use different methods which can make it grow faster. So after it's matured, like it reaches a certain height, you can put it in the soil back again.
to make it healthier and maturity level, it needs certain time and it is a slow growing seed, so it needs its own time. Doing artificial things might even damage the seed and tree might be not.
Method which I'm talking about is not artificial. It's a natural method.
But almond trees requires its own time. You can't force it and do it.
Please, children, please remember, fast growing species are fast dying species. slow growing species live for a long period of time, like our jamun. Jamun is the Nerala Mara. They grow slowly and they live for a very long time. Exotic species, many of the exotic species, they grow very fast and also die very fast. Which one is preferable? You decide. Thank you.
Take up one or two questions from students. Okay, later you can ask them.
Lassia and Clara.
Good afternoon everyone, myself Lassia of 7th standard.
Myself Clara of 7th standard from Andrew Place Girls High School.
So our topic is disposal of unused medicines under waste management. Introduction. We live in a world of diseases and the usage of allopathic medicines has reached an ever-time high. We seldom care about the place where the unused medicines and unused and expired medicines end up. Our search is about how our society deals with the unused or expired medicines. To understand the health hazards of disposed medicines, to identify various methods of safe disposal of unused medicines, to study the effect of disposed medicines on soil, to develop a successful chain of services to assist in safe disposal of unused medicines. Hazards of disposed medicines. If the disposed medicines end up along with the garbage in landfills, animals can accidentally ingest them. This can cause poisoning. Some medicines leach into water systems and spoil the underground water quality. If the disposed medicines end up in water bodies, aquatic organisms can be affected. Pharmaceuticals are reported to be affecting the behavior and reproductive system of aquatic organisms. Drug as simple as Roro 650 can cause liver and kidney damage in animals if ingested. Ask nearby medical stores about drug take-back program. We conducted the stores which were near to our school to know if there is any provision of drug take-back facility. These are some of the questions we asked for medical shops. These are the photos we went to medical shop. Response received. 2 and 3 medical stores told us that they would take back unused medicines if purchased from their shop. All the three medicals were unaware about the existence of any drug take-back program. All the 3 medical stores refused to take back unused medicine purchased from other shops. All the 3 stores replied that they would send the medicines that cross expiry dates back to the distributors, but they did how the distributors discarded them. We also contacted Maruti Medicals, one of the top medical shops in Bangalore, over phone. Unfortunately, even they were not aware about any such system in Bangalore. Survey on disposal of expired medicines among 20 members. We were curious what happens to the unused medicines with the public, so we conducted the survey among our neighbors and teachers. These are some of the questions and options of the survey. Check the expiry date of medicines, always 13, often 3, sometimes 4. Usually dispose of expired medicines. In trash 18, return to pharmacy 4, keep at home 1, aware of any potential risk, yes, level 9, learn about proper disposal methods,
healthcare provider 8, pharmacist 2, allied resource 9, family and friends 14, government compliance 4. Think there is enough public information available? Yes 13, no 3, not sure 4. Will you use a medicine take back program if it is available? Yes 3, no 30, not sure 4. Methods Method of expired medicines disposal preferred. Community take back events 5. Pharmacy return 13. Mail back 3. Special disposal bins in public 30.
Popular pharmaceutical waste management services in Bangalore, as shown in the internet. When we searched in the internet about the popular pharmaceutical waste management services in Bangalore, 8 to 10 organizations were 8 to 10 organizations were reflected. When we contacted them, none of them were connected to pharmaceutical waste management. We also tried approaching BBMP office Nagarbavi over phone. We contacted the following pharmaceutical companies over phone. Team Pharmaceuticals, they do not collect back any old medicines. Microlabs Limited, not picked. We did not find any organization in Bengaluru which took back the medicines, or if such system is available, no one knows about it. We even asked about poor karmikas of our area about the means of disposal of medicines. They told us that they collect the medicinal waste along with the dry waste and just add to the garbage dump. Our studies about the drug take-back program resulted in the following finds. The public does not know about any such system in Bengaluru. Most of the medical shops do not have any idea about how to guide the. Due to lack of facilities and ignorance, people just throw the old medicines along with the trash. Most of the information given in the internet about the organization which deal with pharmaceutical waste management is false.
None of the suggestions by WHO and CDSCO mention or include a proper specific method. Examiner's opinion about the general procedure to be followed by disposing unused medicines. Unopened, unused or expired medicine should be returned to pharmacies or nearby hospitals for disposal, but and no hospital in our knowledge is part of such a program. BBMP's action plan about disposal of unused medicines, the leftover inerts and rejects are transferred to sanitary landfills. The search results are full with terms like suggested, proposed, opined, considered, discussed, etc. Nowhere there was a mention about a proper disposal method. In this digital world, we depend on Google or search engines to find instant details. Our query, how do I dispose unused or expired medicines? Receive the following response. There are sporadic take-back locations in India, most of which are unknown to public. A small study to check the effect of Doro 650 on soil organisms. We took the garden soil as sample, mixed the soil with water and made muddy water, observed the water under microscope to record the microbes. We then added powdered Doro 650 to the same soil and repeated the observation under microscope. So these are the photos of the microbes. We found an actively moving microorganism. We added powdered Doro 650 and after some time found the organism dead. But we know in one or two observations, conclusions cannot be drawn. Some countries with drug take-back program, information obtained through relatives living there. We heard that in Sweden and Australia there are some drug take-back program method. No proper system exists to deal with the waste medicines in our country. Normal incinerators cannot be opted as some drugs may lead to explosion. We add polluting the land with the unused and expired medicines, making the lives of all animals miserable. Chemicals in these medicines can destroy useful soil microbes. No one takes the accountability of this waste. Our project is about to focus light on this serious issue. Recommendation. Medical waste incinerators can be established in every hospital that has 100-bed capacity and a separate wing can be installed in such hospitals for the drug take-back program. The BBMP can keep a specific day in a week for the collection of only unused or expired medicines from the households.
The same can be disposed of at the hospital with incinerator facility. We read that BBP has proposed a solid waste management cess of ₹100 per month for each household. This cess can be utilized for the effective waste management, or some NGOs can be approached for assisting in drug take-back program.
And we would like to express our gratitude for the following individual for giving us an opportunity and helping us in our project. Dr. TVRAMACHANDRA sir, Coordinator of Energy and Wetlands Research Group, IISC, and his team for the orientation program and provided. Our senior Sir Shulka for giving us the idea of this project. The school management and office staff for helping us. Our parents for allowing to participate in the survey. Our teachers and neighbors for participating in the survey. Owners of nearby medical shops for participating in the survey. We also thank our school security for accompanying us to the medical shops and taking photographs. Thank you. These are the references.
Collection of hospital and surgical waste and disposal of them through incineration and other methods. But so far as collection of medical waste, like ulcers, creams and other things, this is really a serious social problem which nobody has addressed. And I thank you greatly for attracting the attention of the society by your paper. And naturally you have stated that in spite of inquiry, everybody gives evasive reply and there is no institution in place for the collection and safe disposal of the medicines at the household level. And this is really a social need. And some people must work for it. And I thank you highly for drawing the attention of at least this member of the society towards this burning problem of disposal of very dangerous disposal of medicines, creams and other household medical.
So nowadays, what is happening is doctors. So these doctors write down whatever is required in the prescription. So if when given the prescription is given to the medical store, they give the exact amount. So most it is not wasted. So why is this required?
Because some people will not use the whole sheet, right? They'll leave the leftover ones.
When giving the exact amount to be taken up by the patient, then what will be left?
Doctors are right, but the patients are not taking care about that.
So we were told that they didn't speak to or and letter to them, right? To aware what is happening actually at your own. Should I repeat?
Actually, he was not having time.
But
Three.
everyone. My name is IISC. Good afternoon, everyone. My name is IISC. I'm studying in 7th standard and I'm from Bangalore Place Girls High School. For today's project, my topic is invertebrates in my campus. Introduction. We were afraid of insects till we learned recently that they do a lot of service to the nature, such as pollinating and crop protection. We also learned about food chain and how each organism play a role in it. We thought of studying more about these small organisms, and it was also a fun activity for us. Objectives: to have a better understanding of an ecosystem, to study the role of various organisms and how they are connected to food chain, to know our surroundings better, to understand the useful and harmful insects, to spread awareness about their importance, to develop photography of these small creatures as a hobby. Study area: school campus and surroundings, materials and methods, logbook and pen, iPad to take photos, twigs and logs for insect hotel. These are the insects which we found in our research. Paper wasp. We noticed that the hive has 6 to 7 cylindrical structures. The color of wasp was reddish brown with yellow band around the belly. This is brown garden snail and that is white garden snail. Gecko. Grey color with distinct white inside. Sorry for the mistake. We was taking photos, so I know that gecko is a vertebrate, but I added to the slide. Crane fly. It looks like a large mosquito, but it has a longer legs. Grey lady bird beetle. Pinkish grey body with black spots and round body. Spider. Black house spider. Legs were dark brown and abdomen was dark grey. Shield bug. Triangular and black triangular mark at the hind end. Earthworm. Long, cylindrical and segmented body, reddish color. This is mosquito. Dragonfly. Black moth-like body covered with fine hairs. Moth. Tortoise beetle. Small helmet-shaped insect. Black soldier fly. Black metallic shiny color. Orange bush fly. Orange color and red eyes. House fly. Red eyes, grey body with black stripes. Stink bug. Greyish brown in color. Black garden ant. Large black head and large abdomen. Jumping spider. Asian ladybird beetle. Bright orange with black marks, a clear M-shaped marking behind its head. Cow bug tree hopper. Brownish black in color, have thorn-like projection on its thorax. Bagworm. Spiral-shaped bag that hung from trees and leaves and branches. Parthenium beetle. Off-white with dark markings. A blue butterfly. Metallic blue markings on their upper sides. Summary. Type of organism and ecological importance. Paper wasp. It helps to control insect pest population and it also helps in pollination. White garden snail. Many animals, including birds, they break down organic matter, improve soil structure. Garden gecko. They help controlling the population of crop pests. They are food source for other animals. Crane fly. They are food source for many animals including birds, spiders and etc. Grey lady bird beetle. They are natural enemies of insect herbivores such as aphids. They are food source for many predators including birds, spiders, etc. Black house spider. They help to control the population of pests like flies and mosquitoes. Shield bug. They are generally considered as pests. Earthworm. They loosen soil and make it aerated. They redistribute nutrition throughout the soil through their droppings. They are a protein-rich source for birds and frogs. Mosquito. They are a food source for many other animals like lizards and frogs. They are pollinators. Mosquito larvae are food source for fishes. Moth. Moths and their caterpillars are food source for many smaller vertebrates and spiders, and they help in pollination. Tortoise beetle. They serve as source of food for smaller vertebrates and invertebrates. Black soldier fly. Their larvae can quickly waste and can help reduce the amount of waste. Their larvae can be used as feed for fish and poultry. Housefly. They consume rotting organic matter and dead animals and thus helps to clean up, and it also helps in pollination. They can control pests like aphids and milts. Orange bush fly. They help in breaking down and recycling organic matter. Stink bug. They are generally considered as pests. Tree hopper. They are food source for insectivorous animals. They have mutual beneficial relationships with ants who protect tree hoppers from predators and drink the tree hoppers' honeydew. Parthenium beetle can be used as biological control in managing invasive species like Parthenium. Bagworms eat damaged and older leaves, which helps to tree grow new, healthy flowers. They are food for small predators. They are their caterpillar that live in protective bags made of silk and trees debris. Oak blue butterfly helps in pollination, food for many small vertebrates. Insect hotel. We learned about insect hotel, made two of them and hung from trees in our garden. Conclusion. It was indeed a fun to observe these organisms, and we learned a lot new things during the observation. These small creatures slightly share our space, but very rarely acknowledge the silence they do to our ecosystem. We came to know that there is a predator beetle of Parthenium, which is a weed. It is called as Parthenium beetle itself. Insects need keen observation, and we experience that this is a can be an excellent hobby. Recommendations. As students, we sincerely suggest that biodiversity observation and documentation can be made one of the activities which is evaluated and given internal marks. TVRAMACHANDRA sir and his entire team of CES department IISC for providing an opportunity to present our project. We also thank our school management and administrations for providing us the support. We would like to thank our parents for encouraging us to participate in such a program. And I would like to thank my seniors for helping me to do this project. Thank you.
Interesting and less studied about animals and insects which are living around us. Thank you very much. It was a very interesting paper. Adali M and Advik.
Good morning, gentlemen. This is Radhavir and
GN
from Vidyaniketan, Bangalore. So without any ado, let's get started.
So our topic is the analysis of Ullar Lake soil. Introduction. Soil is a key resource which helps plants grow. The uppermost layer of the earth's surface is called soil. Soil formation is a slow stepwise process. it takes thousands of years for a layer of soil to form. Soil plays a key role in biodiversity present in an area. Objective. Our main objective is to study the soil pollution, if any, in the soil in and around Ulla Lake.
And also to analyze the chemical of the soil. And growth plays a key role in nitrogen and oxygen cycle, regulates temperature, filters.
The physico-chemical properties of soil. These properties directly influence the abundance and types of microorganisms in the soil, which in turn affect nutrient availability, nutrients and organic matter decomposition.
Here is the geomapping of study area, Ulla Lake, its perimeters and sample sites.
So here are the procedures we followed. That is the collection of the soil samples, dilution of these samples in water, test, pH test, calcium hardness test, TDS, electrical conductivity, and microplastics analysis.
Dilution of the soil in the samples which we collected in and around the Ulla Lake. Then comes the pH test, which helps in determining the condition of the soil.
Next was the NPK test. These are the major chemicals present in the soil, which is nitrogen, phosphorus, and potassium. These nutrients play a key role in soil fertility. So the results are normal for nitrogen, trace for phosphorus, and low for potassium. So let us understand the role played by these three major nutrients. Nitrogen plays a key role in photosynthesis. Phosphorus is essential for cell division in plants, and potassium helps in distribution of water to the leaves of the plant. However, these nutrients should always be balanced and not be excess. So here are the average chemical compositions of soil in Bengaluru. These include the nitrogen, phosphorus, potassium, boron, iron, and manganese. This is the effect of urban activities in soil contamination. These include the industry waste, municipal waste, military, oil production, untreated waste, power plant, transport, mining, etc. Here are the results and discussions. The results of the calcium hardness test, total hardness test, electrical conductivity, magnesium hardness test, total dissolved solids test, and microplastics analysis. Most of these tests are in parts per million or ppm.
Total hardness test is done when the soil is alkaline in nature.
So here is the TDS test. Let us understand the purpose of the TDS test. It is conducted to find the dissolved salts in the soil. It comprises of inorganic salts, calcium, magnesium, sodium, sulfates, etc. TDS levels in the soil can affect the growth of plants. It also determines the soil salinity.
Here is the electric conductivity test. Let's understand the purpose of electric conductivity test. It's an important parameter as it provides the information about the soil's ability to transmit water and nutrients. Electric conductivity is expressed in microsiemens per meter.
Let us come to the most important, most polluting part of the human caused pollution, that is the plastic pollution. this is the microplastics analysis. So let us have a brief about the microplastics. Microplastics are fragments of any types of plastics which are under 5 mm in size. So they are very small. They are very harmful for the environment. Synthetic textiles are the main microplastics by contributing about 35% in the production of man-made fibers such as nylon and rayon. There are two types of microplastics which are primary and secondary microplastics. Every year, about 4 to 40 million tons of microplastics are dumped into the soil and oceans. So we can see here that microplastics are evident in our soil sample. Let us understand the effects of microplastics in soil. Microplastics cling on to root hairs of plants and affecting plant growth. It can also cause oxidative stress to the leaves of the plant. Also, it affects the microorganisms in the soil, which can decrease the soil fertility. So here is the table which includes the results and formula of all the tests we have done.
Here is the contamination cycle which led to the soil pollution.
So let us understand how polluted soil is. Polluted soil containing heavy metals such as lead can enter an organism's body by inhalation or indirect consumption, which in the end pollutes the food web. It can also cause ecosystem damage by causing the ecological imbalance and loss of biodiversity in the area. It can also cause the growth of unwanted in the area. So we can see that there is a certain amount of pollution in soil in Ulla Lake. So this is the waste which leads to soil pollution. We have interviewed 2 agriculturists with one over 55 years of experience. Shri Krishnamurthy gave us the insights that the age-old organic farming is best to maintain the soil health. Also, the pesticides, which are petroleum byproducts, should be minimized. Naveen Kumar, who likes to adopt modern technologies in farming, gave us the insight that adoption of modern technology without hindering the soil health can be manageable without decreasing the yield of crops.
Mr. Ravi and Dr. Jayashree are the board interviewed about their knowledge on the Ulla Lake. Stated that the improper disposal of waste and the idle immersion in the lake can lead to the soil pollution. Dr. Jayashree insinuated that we have to put hands together and clean the lake periodically.
So these are the threats to soil. Recommendations. We recommend the following steps: soil conservation, responsible soil management, support soil-friendly farming, safe waste, and education about soil and its importance to be given to all. Conclusion. Decrease the usage of plastic to get rid of microplastics in nature. Adopt organic farming. Add organic matter which improves soil tilth. Water pollution and other resources pollution should be stopped as they can have an adverse effect on the soil. We thank all of them who have helped us. Thank you.
You started with soil condition in the Ulla Lake area.
Yes, sir.
And then moved on to the soil in general in the Bangalore city. This is not. If your study is about soil condition in the Ulla Lake area, entire not to that particular area.
Sir, Bangalore.
At the end, they have not understood anything about Ullad Lake area, soil condition in the Ullad
Sir,
Lake.
all the tests are done in Ullad Lake area only, sir.
Like the sewage joining the Ullad Lake water. So we considered Ullad Lake.
Ullad Lake, soil condition in the Ullad Lake area different from the soil condition in other Bangalore area.
As Ullad Lake is a lake which has slightly polluted water and soil, it is the soil is slightly alkaline or basic in nature, which means that some macro minerals cannot be absorbed by the plants. So with and also the microplastics maybe from the silt which has deposited from the sewage in the area, as we have collected the soil samples for the microplastics in the outlet and inlet of the Ullad Lake. So the sewage which comes to the lake only and not the Bangalore city respectively, it will differ as the soil in the general Bangalore city can differ and may not have microplastics which have been brought to the soil by the sewage.
Akshara and Vidya, young.
Hello to one and all present here. My name is Vidya M. And my name is Akshara. We are students of class 7 studying in Vidyanaketan Public School, Bengaluru, Karnataka. Today, we are diving into the wonders of Hirikere Lake with the ecosystem of Hirikere Lake, harmony. Introduction. An ecosystem is a community of interdependent organisms which can be they basically communicate with each other and depend upon each other in specific area. Lakes are unique ecosystems which play a vital role in human sustainability. City present in this lake to check the quality of water in the lake. Hirikere Lake was a peaceful lake which had a very good environment but a bit of pollution. Finding a peaceful environment is a great opportunity and being lakeshore. Such one lake was Hirikere Lake. And the perimeter was around 3,666.25 meters. With the materials and methods, we followed strict guidelines given by our teachers, like using gloves and glass bottles. Error method for the identification of bird species. These are the testing kits we have used throughout this process. Total hardness, calcium hardness testing kit from the brand Nice, salinity testing kit, chloride and fluoride testing kits. Physical parameters, the water is collected from pale yellow, whereas the water collected from here. The water collected from all 3 points were tracked.
Fluoride was 0.0 milligrams per liter and chloride was 200. Definitions. Freshwater resources like lakes, rivers, wetlands, and ponds play a crucial role in human sustainability and development. Lakes are facing problems like pollution and eutrophication, which is threat for us. The presence of significant levels of chloride in this lake indicates inorganic pollution. Chloride content was absent here. It is a huge population of birds present around the lake. Ducks, kingfishers, morning ducks, European kingfinch, white-headed eagle, red-whiskered bulbul, brown-eyed vulture, Eurasian spoonbill, and the great cormorant. This is our birds observation. A few of them were engaged in different activities like feeding, flying, etc. These are the insects that we found here: butterflies, millipedes, and dragonflies. So we recommend people to stop throwing garbage around this lake because pollution is the main thing for the destruction. It's a problem in the environment. Not only lakes, many ecosystems are getting polluted due to pollution. And furthermore, I urge people to take part in tree planting initiatives around this lake because they control soil. Would be good for the bird population there. In conclusion, Macroinvertebrate lake provides. In conclusion, Macroinvertebrate lake is important and bring a true change. The references we have taken, and this is us working in the field by collecting samples. We would like to thank the Benny team for Mr. TVRAMACHANDRA for his great support. We would like to thank our school teachers and our Vidyanjali public school. To our parents who support us.
Please tell me whether Kere lake is
Minus of 400 ppm, so it has hard water. It is 9, which means alkaline. So it's not that threat for aquatic species. Because by throwing covers there and we were for students.
so it's neither healthy 50-50.
Yes, sir.
Neither healthy nor
yes, sir.
Why don't you use the power of technology?
So we use the app known as
No, why don't you use the power of technology by reporting it, telling that these people don't listen to you?
So actually we are, my uncle has told that he give the information contact soon. So I'll write an email for sure. That would be done.
Thank you very much. Kumar's from the N, D, and Ulas.
Ulas. Today we will be presenting a presentation on Ulla Lake, identifying issues and propose solutions. Lakes play an essential role in maintaining ecological balance. Lakes shelter many species of microorganisms, plants, animals, and birds. And lakes with specific and significant plant life act as a carbon sink, helping climate change. And lakes also replenish the groundwater levels. Our objective is to analyze the current state of Ulla Lake by testing its physical, chemical, and biological parameters and to enhance the condition of Ulla Lake, and to propose cost-effective, nature-based solutions for its preservation and rejuvenation. Study Ulla Lake, located in the southwestern part of Bangalore city within Ulla village, located in the Vrishabhavati valley, coming under the Bairamangala Lake series, covering up to 11.03 hectares with perimeter approximately 1,870 meters. These are the 3 sample sites from which we took the samples, and we for comprehensive analysis. Materials needed. The materials 3 compound glass containers, various water testing tubes for parameters, and a microscope for microbial analysis. We collected the samples for totally 8 hours by collecting them. We then tested them for various parameters such as pH, dissolved oxygen, and hardness. And additionally, the samples were observed at a microscope to identify any microorganisms. Findings of Ulla Lake. The first picture shows the different plants created to separate the lake into 2 parts. The third and the last picture shows us collecting the samples and performing the on-site parameters. And the fourth picture shows the outlet, and the fifth picture shows the inlet. The physical conditions of Ulla Lake include color, which was watercolor, which was a pungent smell. Floating plastic and algae blooms were particularly visible at outlet, indicating pollution. Chemical parameters. The pH 8 to 7.2 across the sides, and the total hardness of the water varied from 235 milligrams per liter to 192 milligrams per liter. The calcium hardness highest in the sample site number 1, which is inlet, and the lowest in sample site number 2, and magnesium hardness varied from 70 milligrams per liter to 56 milligrams per liter. The chloride levels was highest in sample site number 1 and lowest in sample site number 2. The fluoride was absent in all 3 sites. Alkalinity varied from 190 milligrams per liter to 175 milligrams per liter. The DO varied from 5.03 milligrams per liter to 5.27 milligrams per liter. The biochemical oxygen demand in the water varied from 0.53 milligrams per liter to 0.25 milligrams per liter. The electric conductivity varied significantly. Varied from 573 milligrams per liter to 245 milligrams per liter. Microbial analysis from the presence of Anasystis, Adeno-cyanobacteria indicating purification, which is a process driven due to excessive nutrient levels, leading the pH was alkaline across the sites, which has accepted water quality, but the calcium hardness and the DO was moderate, supporting some aquatic life, but raising concerns about the
organic matter, and the biochemical oxygen demand was very low, indicating minimal decomposition of organic matter, and the fluoride was absent in all three sites. The fluoride was absent in all three sites.
Issues. The primary issues we found in Ulla Lake are sewage contamination, untreated sewage water is flowing into the lake, leading to pollution. Illegal encroachments are reduced in this area and worsen the pollution problem and adversely affected especially bird species.
Which are recommended reintroduce building artificial wetlands near Ulla Lake to filter this untreated sewage water which is left into the lake directly using microorganisms such as Bacillum subtilis and utilizing aquatic plants such as observe the excessive nutrients such as nitrate present in water, installing solar-powered aerators to increase the DO levels to support the aquatic life present in the water, and reintroducing native fishes such as ruhu to control the. These are the references which we took to research. I would thank the Lake Symposium team and the IISC members for giving us this opportunity to express our research. Also thankful for our school, our director, vice principal, and our teachers, and especially our mentors and students who have this project. And a special thanks to our parents for this project.
Why untreated sewage is entering into the lake?
Sir, actually, the BBMP had planned to filter it, but they have also built a filtering plant, but due to their irresponsibility, they have not even started it, and the building is not even completed.
There is no sewage treatment plant. Plant is not working, the street is not at all working.
Yes, sir.
Come on, half completed building is there.
Question. What is the government doing, or is the government doing anything to registration?
Actually, recently, there was an article published in Prajavani on 9th October. The officer had mentioned about Ulla Lake being clean.
Secondly, it is Siddharth page and
Good afternoon, everyone. My name is Havan from class 7 from Invertebrate.
I'm Siddharth.
I am Samanthi, and today me and my friends would like to share with you our presentation.
Today we are going to talk about the crucial ecosystem that is often overlooked but is incredibly vital to our planet, wetlands.
Wetlands are vital ecosystems that bridge land and water, creating a diverse array of plant and animal wildlife.
And the wildlife we have chosen to speak about is the Malathamadi lake.
The main objectives include emphasizing the material benefits humans derive from natural ecosystems, looking at the non-material benefits ecosystems provide to human society,
Learning the material benefits that ecosystem directly provide to the human community,
and the benefits that ecosystems provide by regulating natural processes and maintaining environmental and
study area included Malathamadi freshwater lake, which is located in the Vrishabhavati Valley, spreads over a large area of 22.36 hectares and comes under the south ward of the wetland.
method we use to identify ecosystem services is by mainly observation and topic.
We visited the lake on 3 days, observing various things, and we made sure that we utilize all these 3 days to their best. On the first day, we mainly focused on the observation and identification of bird species, which we also continued doing on all 3 days.
second day we mainly concentrated on observing features of
Last day we concluded by working on cultural and coordinating skills. We also utilized our time to explore other things. So, ecosystem services. Ecosystem services are
and regulating and supporting services.
The concept of cultural service refer to the non-material benefits that ecosystems provide to the human community, including a wide range of values deeply in aesthetics, cultural identity, and recreational activities.
oceanic services of a lake refers to the material benefits derived from natural ecosystems.
The regulated service of a lake regulates the natural processes and maintain environmental quality.
While explaining the concept of cultural service, first comes connecting people to nature. Lakes have a unique way in which they connect individuals to nature and its beauty by capturing their minds and serve as a source for tranquility, inspiration, and creativity. Lake grounds. Malathali Lake grounds holds cultural and social importance within the local community. Science and education. Lots and lots of people come to the lake for bird watching and photography. They see people who share the same hobby of which they follow. Next, sense of place. When we spoke to the people there regarding biodiversity and how they felt about the surrounding area and what changes they wanted in the lake, they answered with a great sense of place and they are proud to belong to a community which house such a great lake.
The provisioning services of the lake. Water supply. As we observed no outlets, we can conclude that there is no water to the surrounding society. The main inlet was rainwater. This lake played a really important role in the local identity. From no abundance of fishes, not all, we can conclude that there was no advantage of water, advantage of fishes. We found basic plans for the domestic humans.
The important quality we observe that the dragonfly about the lake, and we also interviewed them if the government had done some changes and how they felt about the walk paths there. Their replies were general. Their replies the walk paths had no effect to the biodiversity there.
Further research on the birds of cat are birds that is good, grey heron, white-throated fishes, spotted red-backed flathead, cormorant, black kite, Indian pond heron, white-breasted, Indian ecological, gargantic duck, peacock, painted stork, and a moth.
That's it. That's the.
Observed a large area of encroachment of land has been done, and there is no significant effort from the people to reduce plastic in the lake grounds. And it is littered, and there are no dustbins in and around the lake. Next, we have IUCN red list, and which are least concerned. This we can conclude that Malaysian lakes are significant cultural resource, offering a range of cultural values that enhance community well-being, provide aesthetic transport, and support environmental education.
Though the provisioning services of Malagali Lake does not provide much, it acts as a significant
In metropolitan city like Bangalore, Malathali Lake sure helps regulate and maintain a healthy ecosystem for all organisms. We identified about 14 bird species.
Is that don't press it? No, they're pressing it and talking.
Everyone who has an interest in this project, these are the references. Thank you.
Development Lake is providing only recreation and protection.
is providing but not to create
We have observed that people, instead of utilizing the peace of the lake and enjoying the nature, still a huge area of encroachment lake ground. It is converted into a ground, and people come for activities such as playing cricket, jogging. They don't usually come around the lake area. The lake area is almost
isolated. So only few people who are interested we only saw people who were interested and very few people who were interested in the nature.
So we can greatly enhance
No, actually, sir, the lake has been regenerated and sir, recently they have installed like half of the lake has been and get down to the lake. Still, we saw plastic lying around the lake. It was floating upon the river at the pond. And still, lots of area itself, construction. And the birds, every time they come to rest, the sound that the vehicles create, they fly off. They don't have peace there. So people should take access to this. But it's kind of, as you told, they only come for activities. Thank you, sir.
मौखिक आध्यात्मिक
We are going to present our research and impact of Ganesha idol immersion. Biodiversity public school have investigated into the Ganesha idol immersion on Ulla Lake's water quality. Body of still water surrounded by land on island. Lakes are an integral part of the ecosystem. Lakes are crucial for many reasons, including maintain a healthy ecosystem, for recreation, for the groundwater, and the understand and analyze parameters which determines the water quality and after the immersion of ions in these freshwater bodies. We have conducted a study of Ulla Lake. It is the Ulla Lake, the southwestern part of 3 acres, the total area of 3.5 acres square. These are the materials used for conducting. We followed throughout our project. First, we went to the site then we conducted many kinds of physical and chemical parameters in compound microscopes.
These are the procedures to perform our chemical parameters.
Let us see to some of the physical parameters that we check for. Color. The color of the first sample collected before the immersion of idols and the second sample that was collected after the immersion of idols, both were green. The first sample was transparent, where chemical parameters. The first test that we performed was the pH test, where our results were 7.2. We got our results as in the first sample and 130 mg per liter in the second sample. Next, we performed the fluoride test, where we got our results as 0.0 mg per liter in both the samples. Then, we performed the alkalinity test, where we got our results 65 mg per liter for the first sample and 500 mg per liter for the second sample. We also when our results were 192 mg per liter for the first sample and 248 mg per liter for the second sample. Next, we tested for calcium harness, where our readings was 150 mg per liter and 185 mg per liter respectively. Further, we tested for test, where we got 0.0 ppm as our reading in both samples. Next, we tested for TDF and the stored weight dissolved solids. We got 219 mg per liter in the first sample and 460 mg per liter in the second sample. Further, we tested for electrical conductivity, where we got a reading as 448 microsiemens in the first sample and 906 microsiemens in the second sample. Next, we tested for magnesium hardness, where we estimated our value as 42 mg per liter in the first sample and 63 mg per liter in the second sample. Additionally, we tested for DO and DOD, where we got our readings as 5 mg per liter in the second sample for dissolved oxygen. For BOD, we got 0.04 mg per liter in the first sample and 0.08 mg per liter in the second. Lakes, wetlands and ponds are among the vital freshwater bodies that support daily human activities and provide indispensable services. These water bodies is deteriorating, threatening biodiversity and human health alike. Research shows that 80% of the diseases in developing countries are linked to poor water quality. Though these water bodies are affected by natural processes such as rainfall and erosion, the growing human population coupled with developmental activities have greatly escalated the deterioration of water quality in the form of industrial, domestic and sewage disposal as well as agricultural runoff. Key parameters accessed during our study includes the water temperature, a pivotal parameter which directly or indirectly influences numerous other water parameters. Electrical conductivity, a measurement which measures salinity, ionic strength, and solute concentrations. TDS primarily constitutes of salts like calcium, potassium, magnesium, and many more. The hardness of water primarily attributed to calcium and magnesium ions. The pH, a measure of acidity or alkalinity of water which makes it perfect for use. A crucial factor in aquatic ecosystem, serving as a vital determination of survival of the aquatic life. BOD serves as a metric quantifying the oxygen consumption by aerobic organisms during decomposition. It has provided significant changes in these parameters after the immersion of Ganesha idol. There was an increase in TDS, hardness, and POD values. There was also a decrease in DO levels. The significant changes in chloride, POD suggests water quality deteriorated after the Ganesha idol immersion, which emphasizes the urgent measures to protect Kula Lake for the future generations. Let us see some of the issues faced by Ulla Lake. Encroachment. Human activities in and around the lake, like immersion of idols and construction and other activities, may deteriorate. Unregulated discharge of untreated sewage into the lake can increase the nutrient levels and decrease the oxygen availability, harming the aquatic life. By this, many of the aquatic and plants and animals may get endangered. Solid waste dumping. Disposal of waste in and around the lake can block the water flow, reduce storage capacity, and increase contamination risks.
Some of our recommendations include creating public awareness and do not immerse in chemical idols in the lakes with the immersing chemical idols in the lake, but using mud idols as an eco-friendly alternative.
Conclusion, Ulla Lake's water quality is deteriorating. This is the interview that we conducted with our neighbors. Questionnaire where it consists of the question and answer that was there in our interaction. Ulla Lake's water quality is deteriorating. One of the major reasons is idle immersion. Collective efforts can restore the lake's water quality for a sustainable future. I would like to thank the IISC team and the throughout our project. We would also like to thank our friends and other participants who have encouraged us. Thank
Generation. We would like to conclude this presentation stating the quote that we need blue to protect the greens. Thank you.
Paper, what alternatives do you suggest for the immersion of Ganesha idols?
Ganesha idols made up of mud, paint, as paint contains chemical which cannot, and the sewage wastewater treatment plant cannot, the water cannot be purified in a wastewater management treatment plant due to the chemicals present in the paint. So we would like to have a Ganesha made up of completely mud without any chemicals and without paint.
So when we celebrate festivals, like it's because you need to pray and we need to get blessings. It does not matter that will we use chemical idols or idols made of mud. That is the main recommendations that we would like to do. Questions?
Ganesh and Yashas.
One and all present here. Myself, Yashas.
And myself,
We are here from Vidyaniketa Public School. Today, our topic is plankton biodiversity and biomonitoring of Kingley Lake. Let's start.
Introduction. Lakes in Bengaluru. Over the past years, there is a severe growth in water pollution of Bengaluru due to untreated sewage entry and dumping of solid waste. Examples, Bellandur and Vardur Lake. I can clearly state that without hesitation, this is clearly due to human negligence. Encroachment. There is a massive growth in the urbanization in Bengaluru over the past decades, where the construction of buildings in playgrounds and encroachments of lakes, loss of interconnectivity. In the past, we had a well-versed water treatment, lake water treatment a system which was known as Rajakalugas. Now, due to the destruction of it, we have lost the proper lake water management system of Bengaluru. These are the conservation efforts put forth by the society to conserve the lakes.
Bangalore once had a well interconnected lake system. It, the system was damaged by encroachment and frequent urbanization. Who is responsible for this? The citizens or the government? All of them, all of the people who are there in Bangalore are responsible because people think urbanization and improving technology is the main goal, but they don't think about environment. And we can see the consequences. Frequent floodings are happening in the past due to clogged sewage systems. We can see frequent water shortage, which happened in the month of March this year. We also see environmental damages. The lakes suffer from habitat loss, and animals lose their homes due to encroachment, silt deposit, and unchecked constructions.
And this is due to loss of interconnectivity. Objectives: to analyze and measure the physical and chemical parameters of the lake water, to identify plant and biodiversity of the lake, to create awareness among the community people about lake water pollution.
This is our study area. Kengeri Lake was also known as Kengeri Lake.
Located in Bengaluru Kengeri satellite town, it spans across 25 acres, constructed by the State Forest Department of Karnataka in 1995 at a cost of 95 lakh rupees, includes islands, pathways and fencing. Current condition of the lake is it overruns with weeds, it's discolored, covered with hyacinth and a breeding ground for mosquitoes.
Materials and methods. First, we need a standard water testing kits, compound microscope, lake water samples, and digital mapping. This is the study area, the dimensions of Kengeri Lake. The Kengeri Lake area is 66,625 meters square, and the perimeter is 1,520 meters.
In this slide, we can see two images, one from 2012 and one from 2023. We can see a clear difference between both the images. We can see a lush greenery in 2012 and in 2023 we can see complete desertification. Here we can see lake water is present, but here it is a barren land. Here we can see plantation. If carefully observe, there is a rapid urbanization over the past decade.
Here, if we see, we can see the latitude and longitude where the lake is located.
The encroachment images at the bottom. Reports upon lake. Kengeri lake water is black in color. There is improper fencing and there is huge macrophyte covers overall. Problems is untreated sewage entry, dumping of solid waste and building debris. Improper fencing, massive growth of macrophyte.
Here we can see the water status. The water is completely eutrophicated.
We can see plastic pollution throughout the lake shore.
These are the points where we collected waters for samples. These are the dates and times when we collected the samples.
These are the water samples, and this is the way of retesting the water samples. These are the results of the after testing the water samples. In total 5 tests, which is alkalinity, total hardness, pH value, calcium hardness, and dissolved oxygen, and the readings are in the table. Results and discussion.
In point 2, as we can see, alkalinity is high and alkalinity can negatively affect lake water in many ways. Like example, increased pH is harmful for aquatic life, plant growth, and chemicals may precipitate on pipes and clogging of pipes may happen frequently. We can also see high total dissolved solids. This can cause cloudiness, discolor, and unsuitable for human consumption. It disturbs ecosystem by harming the wildlife and reduces oxygen levels, leading to low productivity in nearby areas.
In lakes can negatively impact the aquatic life by damaging the skin and gills of the fish. It can increase toxicity of ammonia and other harmful substances. This leads to eutrophication, excessive growth of algae, thereby reducing the oxygen levels, scale formation, damaging the equipment.
Now let's see the planktons we observe.
Trachylomonas. Here
we are able to see a small eudorina.
This video of plankton observation. The first one is the top one is tabullaria and the bottom one is brachiones. This was a video of an unidentified object. Pros of planktons to the lake. Pros of planktons in the lake. Planktons, especially phytoplankton, generate oxygen through photosynthesis. Planktons are the base of aquatic food chain, supporting fish and other wildlife. Nutrient cycling. Plankton help recycle nutrients, maintaining the lake's ecosystem balance. Cons of plankton in a lake. Excessive plankton, especially certain algae, can cause toxic blooms, depleting oxygen and harming aquatic. Water quality issues. Algal blooms can make water murky and unsuitable for human consumption.
Now let's look forward to the recommendations we gave for lake engineering. After examining and understanding about the lake, we came into a conclusion that we used two methods, hydroraking and bioremediation.
Hydroraking is a method to remove aquatic vegetation and sediment in lakes and ponds. It's completely environment friendly. This process is hydrorake mounted on a paddle boat to scoop up and benefits. It preserves ecological balance and improves recreational value in water bodies affected by urbanization and climate change.
Bioremediation is a process that uses plants, microbes, and other organisms to degrade pollutants, contaminants, and toxins from soil, water, and other likely environments. There are two types of bioremediation. Nanobioremediation. This kind of bioremediation uses nanotechnology, including enzymatic degradation. Microplant-based remediation, it combines the strength of microbes and plants to degrade pollutants and restore environments effectively.
Inference, Kengiri Lake is polluted due to waste dumping, sewage and excessive microphyte growth, classified as Class C by Central Pollution Control Board. The water is suitable for irrigation and we can observe macrophyte cover and zooplankton. Conclusion, the lake is heavily polluted, macrophyte cover is observed, the water is unfit for human use. We would like to thank everyone who supported us in this presentation. Thank you. Thank you.
The recommendation we have omitted one important thing. Export all your polluting lake waters to Kolara. The government is doing the same thing. Yesterday this project a lot about it. Yes. All the equipment is being exported to Kolara and send this
billions of those.
And the water, that happens when the water is flown excessively. The excessive water, what happens when the water is waste, it flows out, and people are not able to control the water. At that time, they collect the water and they export it to other places, which is not treated as you said. Question. Planktons are organisms similar to algae, which live in which are kind of aquatic animals. Organisms. They are kind of algae.
For collecting water samples, we selected would be step to the 4:59 5 o'clock. We select only 10
It was
Because the next day we needed to go to for school to testing it. So as evening it would be, so the the temperature of it wouldn't change in a month.
And to test samples, you need to preserve it. So rather than taking a long time to preserve it, we had collected it in the evening, so it would be accurate measurement.
Thank you.
With Sri Harshal and Ramya.
Respected chair, dignitaries and audience. I am Tana Muralidhar from Vidyanikedhan Public School, studying in 8th standard. And today we are here to present our paper, Preliminary Studies on the Determination of Water Footprint for Spinaceoleraceae, Trigonella phenocrystium and Coriandrum sativum plants. Water plays a vital role in a plant's life by helping it photosynthesize, maintenance of cell turgor, and acting as a transport medium. Water footprint is a comprehensive measure of the total volume of water used in the production of all goods and services, while the water footprint of plants is especially the measure of total volume of water used in the production of optimum yield. Determining the water footprint of plants is the main goal of this study. Why have we chosen spinach, fenugreek and coriander for this study? You won't believe me, but spinach, fenugreek and coriander are three of the most nutritious green leafy vegetables eaten in India. Objective: to determine the water footprint of green leafy vegetables, Spinaceoleraceae, Trigonella phenocrystium and Coriandrum sativum, and to recommend strategies to conserve water and obtain optimum yield for the three species of plants. Study areas. These are the three study areas where we have done the study, and all of these three areas are located in a 1 kilometer radius. These are the materials which we used for this experiment, and some of them are polythene tent, which was used to protect the plants from external water such as rain from entering, and filter pH paper, which was used to determine the pH of the soil and water. This is how we determine the water holding capacity, and we have used the above formula to determine it. Soil was prepared with manure in the ratio of 3:2, and the seeds were sown and taken care of.
Physical and chemical parameters. Water holding capacity Soil pH was 6.5. Soil texture was coarse mixed with fine soil particles. Soil type was loamy. Water pH was 7.0. Manure type was dry cow dung manure. Methodological data. The temperature varied between 20 to 25 degrees Celsius and the humidity was around 76 to 81%. Treatments. The treatments were aimed at water stressing the plants by providing known quantity of water at periodic intervals. Each treatment had 3 replications. T1, T2 and T3 had 10, 20 and 30 ml of water every day. T4, T5 and 10, 20 and 30 ml of water alternate day. T7 had 60 ml of water every day and T8 was drip irrigation. Additional treatments of 40, 50, 70 and 80 were done with spinach. AL is an abbreviation for alternate.
Observations. Observations on growth, health and productivity were assessed to study the plant's response to the treatments. Observations were made on 10, 20, 30th and 35th day post-treatment. The crops were harvested on the 35th day, quantified and its productivity was not here. The one which is submitted. The one which is submitted had all our graphs and others. Results. Plants watered 10 to 20 ml on alternate days gave us a productivity of 11.42 and 14.29 grams per liter of water, which when compared to the 60 ml everyday water, the productivity was very low. And even with 30 ml of alternative watering, we did not get the sufficient amount of yield. So by looking at these images, we can infer that alternative watering is not conducive for this plant. Results of everyday watering, 10 to 30 ml watering every day gave us in results until 15 to 20 days, but the plant started to retard in growth and became stunted. Their leaves were less succulent compared to 60 ml every day. The productivity was more than alternate days plants, but less than 60 ml every day. Hence, by looking at these images, we can conclude that 10 to 30 ml watering every day is good for the plant, but only until 15 days, and we need more water for the plant. Results of drip irrigation. Drip irrigation only gave us 2.14 grams per liter of water as the yield, and hence drip irrigation is not good for optimum productivity. And what plants water 40 to 80 ml every day have grown leaves with broader and larger elongated succulent leaves. However, the plants watered 40, 50, 70 and 80 ml did not give us much better results than 60 ml every day. Hence, we can conclude by looking at these images that 60 ml every day is the best and 24 drip irrigation is the worst. Attempts to increase or decrease the water input would give us lesser yield. Coriandrums are type of productivity chart. The total water footprint of this plant was 22.30 grams per liter of water. Here we can see the alternative plants watered, and the grams yield was 11.4 to 11.5 and 12.82 grams per liter of water. The everyday plants was 15.4 to 17.04 and 19.04 grams per liter of water. And here, if we compare these two graphs, then we can say that alternative watering gave us very much lower yield compared to everyday watering. And here, by looking at this result, we can even conclude that alternative watering is not conducive for this plant. Everyday watering of 10 to 30 ml gave us good until the 15th day, just like how the spinach oleracea plant was. And when after that, we need more water for this plant. Hence, we can conclude that everyday watering until 30 ml is not sufficient for the plant. 30 ml every day is compared to 60 ml at drip irrigation, and drip irrigation gave us very low yield. Hence, we can conclude that drip irrigation is not suitable for the plant, and every day is the best out of all the experiments.
Trigonal phenomenon productivity chart. The total water footprint was 24.02 grams per liter. The results for alternative watering was 12.17, 11.1, 11.59, 13.67, and the results for everyday watering was 11.18, 15.4, 19.44, and 24.02. The results for drip irrigation gave us only 5.83. From here, we can get to know that the highest results was 60 mm, which was watered every day, and the lowest was drip irrigation. The results for alternative watering. The plants that were watered alternate day gave us a poor survival and had a very stunted growth. Hence, we can conclude that the alternate day watering is not suitable for the crops. Results for everyday watering. We can see that the everyday watering gave us a much better results than the alternate watering was sufficient for the plant. So also we can here conclude that the everyday watering is also not suitable for the plant growth. Drip irrigation, which was supplied for 24 hours, which was 240 ml of water, this gave us the least amount of optimum yield, and which is not suitable for the plants. Hence, we can conclude that even drip irrigation is not suitable for the plant growth. Thereby, we can conclude by this project that the 60 ml of water worked with all the three plants and gave us the optimum yield.
Conclusion. From the result of this study, keeping all the influential factors constant except for water, we can conclude that the growth of the three species of plant is governed by the quantity of water offered. It can be noted that spinach could produce green leafy vegetables of 27.78 grams per while coriander and fenugreek could only produce 22.30 and 24.02 grams per liter of water. However, considering the recommendation for spinach, the optimum productivity might stand at 32.07 grams per liter of water. So it may be noted that any attempt to increase or reduce the water input would lower the chances of health and productivity of these three plants. This finding helps us to conserve water by optimum yield. Providing any excess water will result in water wastage, and at the same time providing lesser quantity of water results in poor growth. However, it should be noted that these results are in nature and need to be studied on a larger scale involving different seasons. Plant growth and productivity is influenced by many factors: soil type, soil nutrition, pH, etc. These are the references we referred to while doing this experiment. We would like to thank IISC Symposium team TVRAMACHANDRA for this opportunity to work on. We are indebted to our school and the teachers who gave us guidance and encouragement. We thank our parents for their support. Thank you.
90% of the available water in the country is used for agriculture purpose, and there is enormous amount of wastage of water in wrong kind of irrigation, which is called flood irrigation. And your paper, I really appreciate your paper, because you have drawn the attention of the people towards economizing water and in order to get better productivity of our crops. This is a very crucial area for the country now in the days of climate change, because water scarcity and drought conditions are repeatedly appearing in the country. So from that point of view, your experimentation and your paper is highly commendable. Thank you.
Thank you, sir. Sorry, my teammate here actually lost his voice because he drank cold water yesterday, but because of that he couldn't present today.
Cold water or ate ice cream?
Drank cold water. Any questions from the audience? Thank you very much.
SB, Prakash, and KB Aditi.
Good evening to all present here. Myself KB Aditi from grade 8 Vidyaniketan Public School, Bangalore. Our topic today is soil pollution and its adverse effect on mankind.
Soil pollution is causing major issues to humankind and coexisting organisms. This is mainly caused due to industrial let-offs and improper waste management. Plastic is contributing to major parts of soil pollution.
The main objective is to compare two soil samples and identify the presence of microplastic in them.
Totally two soil samples were collected. One of the soil sample collected was from a BPMP park and the other from a land near Vishwanath River.
Materials and methods. The soil parameters observed include physical parameters and chemical parameters. Materials used are soil sample, universal indicator, filter paper, sievers, NPK testing kit, microscope, Surantri ethyl alcohol.
The physical parameters conducted were water holding capacity and texture. The chemical parameters conducted were pH value testing, NPK testing, and microplastic reaction.
Contamination of soil by enormous concentration of toxic substances. It is caused by both human and natural causes, but over time, human ones are dominating over the natural causes. Factors contributing to soil pollution in soil collection site, as per our observation, are local industrial activities and improper waste disposal.
Here we have tried analyzing the place from where the soil from garbage dump area was collected and the components found are listed.
To illustrate our intent, we collected two soil samples, one from a park and one from a place where plastic was dumped for a longer period of time.
The first physical parameter conducted was water holding capacity. The water holding capacity percentage of soil in the park is 120% and the water holding capacity of garbage dump area is 80%.
Soil texture was calculated by weighing 130 grams of soil and sieving it using the standard sieve for about 10 minutes, and the results are in the slide.
Next was NPK testing. For this, the soil and water was made in the ratio 1:2 and the results are listed.
Microplastic identification. We added the soil solution to solution of Surantri and ethyl alcohol, and we did find microplastic in the soil which was collected from a plastic dump place.
And discussion. The water holding capacity of the soil from the park is more compared to that of the soil from the garbage dump area, and both the soil samples are highly alkaline, and the NPK of soil from the park is not sufficient, and the NP from the garbage area is not sufficient, and the microplastics from garbage dump area is
Microplastics on soil biota and ecosystem processes. Microplastics are small plastic particles which are found everywhere. These can affect they can affect the biomass and fertility and decrease aeration in soil, affect the human health through bioaccumulation of microplastics.
The soil from the garbage dump area was predominantly polluted with milk packets. Some were containing some amount of milk.
which we tried finding out microplastic presence in them, and also we wanted to find out if the plant Argus hypogia, the roots of this plant will take the microplastics from the soil. Hence, we planted the plant and used the root solution to find out if they uptake the microplastics from the soil.
And the research showed that all three substances had microplastic in them. Additionally, the consumption of plastics by cows can result in the presence of microplastics in that cow's milk, which we consume, which affects human health.
Plant has the ability to uptake the microplastics. The roots of the plant Argus hypogia has the ability to uptake the microplastics from the soil. Soil from the garbage dump area has more phosphorus than potassium compared to that of the park area. The water retention capacity of the soil from the garbage dump area is less compared to that of the park area. pH of both the soil samples are almost same, alarming microplastic biomagnification through microplastic testing.
Recommendation: Idianagus crisis is a bacteria that has an ability to degrade microplastics in soil. People can adopt a proactive approach in monitoring and managing waste, make use of 3 Rs, and educate communities about the same.
Like to thank TVRAMACHANDRA sir and the Lake Symposium team for giving us this wonderful opportunity, and we are indebted to our school, our teachers, and our parents for supporting us throughout this journey.
Food articles, they are everywhere. You can also think of and talk for discussion in the later area about biodegradable plastic. That is popular. It is made out of especially this palm starch and also a good alternative for this plastic pollution. Read a greater detail, present effect in the later. Abhi, Arnav, Kishore Magali, Vedant Vikas.
Start. Exposure to natural environments have been proven and shown to be to show calmness in humans' life, especially when you breathe in that fresh air. Don't you feel fresh? Exactly. But what if in an area 2,500 people, more than 2,500 people, for only 6 hours are stranded there with only 350 trees? This situation raises a question in which are the oxygen produced by these 350 trees enough for the 2,500 people? To find the answer for this question and many other, a team comprising of myself, Arnav,
Myself, Aditya, myself, Vedant.
have found and viable answers. So first, the objectives. Objectives of this project are to find the oxygen produced by the trees of our campus and to measure and analyze the oxygen consumption and carbon dioxide production from different grades and the teaching and non-teaching staff from our school. We get oxygen from trees and we exhale carbon dioxide. And the carbon dioxide is used by trees in the process of photosynthesis for food making. That is called ecosystem services. In general, they are the benefits that nature provides us humans. In simple words, they are things that ecosystem for humans, such as resources, regulation, support, and culture. This is the study area where we have done our experiment, and the ground area is 26,950.85 meters square.
Obviously, the materials we needed and the materials, the methods we followed. So, the materials we needed was a pen, a paper, a notebook, and a mobile phone for identifying the trees, and a calculator for finding the value. And the methods are, at first we surveyed the number of trees and their types in our school campus, and then we use standing calculation methods to find the oxygen consumption, oxygen production, and consumed, and carbon dioxide produced by the staff and students of our school. And then we acquired information about the height, weight, and number of students in each grade. And then we calculated the average weight and height from the above data. This is the flora of Vikaniketan Public School, and this is us working.
Oxygen production by trees. Photosynthetic processes in trees result in oxygen as a byproduct. The oxygen released by Vikaniketan trees can be calculated by using oxygen production method, that is leaf area into photosynthetic rate into light hours, whereas photosynthetic rate is 15 micromole meter square per second, and light hours is 6 hours. 6 hours. This is how you calculate the leaf area. Example: how to calculate oxygen production, and we took an example of mango tree, and we got the total oxygen production as no. And the total oxygen in liters per day is 274.31 liters per day. Excel spreadsheet where it shows the oxygen production by different tree species in our school. And this is in the bar graph where it shows the most oxygen production trees in our school.
Now we'll see the oxygen consumption by the people of Vikaniketan Public School. To find the oxygen consumption, we calculated the BMR, and for calculating the BMR, we needed the weight and height and number of students of the grade. This table is with the grades, their weight, their height, and number of students in particular grade. We used the following formula and calculated the total oxygen consumed by the people. For example, we did for first to fourth grade, and we got the total value as 2,34,915.975 liters per day. The unit is actually wrong. We updated in the new PPT, but it's not updated here. And similarly, we did it for 5th to 7th, 8th to 9th, and 10th to 12th grades, and we got the following values. And now oxygen consumption by teaching staff and non-teaching staff. We did the same formula and got the teaching staff value as 54,698.8600 liters per day, and non-teaching staff as 1,228.1713 liters per day. So total oxygen consumed by staff and children is 7,41,770.3213 liters per day.
Now we come to the final subtopic of our main agenda: carbon dioxide production from every people in our school. So for this, we use the formula in which we multiply the volume of oxygen and respiratory quotient. So the respiratory quotient for mostly everyone based on their diet, it will vary, but we have taken the average at 0.85, and the volume of oxygen in Bangalore and in our school is 50. So for one student, you will get 42.5 liters. So that was just an example. Now we'll come to the real thing where we show for each grade and we multiply with the number of students there. So this is until 6th grade, and then we go up to 12 and the staff. Next we have, finally we have the total, which is 147.28 liters. That is how much carbon dioxide is emitted from us.
Final ratio. In this research, we found the final ratio of oxygen produced by the trees in Villaniki and oxygen consumed and our carbon dioxide output is 2,34,500 to 7,41,770.3213 liters is to 147.28 liters. The same has been shown in the bar graph.
Here we can conclude that our school is oxygen deficit, where the oxygen consumption is more than the oxygen production. And during the pandemic time, we used to pay for oxygen cylinders which had 250 liters of oxygen. So let us conserve the naturally available oxygen.
Now we come to the suggestions that we ourselves are giving. In the name of afforestation, we cannot plant random trees in random places. So for this, we use a specific thing, which is plant canopy trees. They have broader branches and more in number, which we can use for more oxygen production. Then we have green schools for better ecosystem environment, in which we take initiatives to promote afforestation. So eco warriors, personally I have planted more than 15 saplings last year. As an eco warrior, there were many in this room from our school. Then we have plant as many trees as possible around the area. Many areas around our school where plants were there, they were removed and construction was happening. So instead of that, we can plant it in our own place and around the area where we still can do it. And create artificial water bodies. There we have some space in our school where we can do this, where we can plant marine plants which also will emit oxygen. Vertical gardens, hydroponics and aquaponics, which are modern methods in which we can save space and also still plant trees. The references that we use here is our own school website, many search engines and AI bots, books such as Botany of Life of Plants, CO2 emission mitigation, energy, and plant physiology and development. Then we have websites such as ResearchGate and Carbon Footprint, and apps to identify the tree type. We have used plant.
We would like to thank the IISC Lake Symposium team and Sir TVRAMACHANDRA sir for this opportunity and platform to welcome.
We are indebted to our school, Vikaniketan Public School, Woodland, Bengaluru for the continued support provided.
We also thank our teachers, especially our in-charge teacher Usha ma'am and teachers Mangla ma'am and Meera ma'am and Pramod sir for their encouragement throughout our project.
We also thank our parents for the support in this project.
Questions?
Communication is oxygen deficit. This is not at all a good news for us. And you must improve your school campus at this particular. So, your oxygen deficit, in the sense, you are borrowing oxygen from the neighboring locality. You owe oxygen to those or reduce your carbon dioxide emission. One of the two.
Sir, in the coming days, we would like to plant more trees in our school.
Thank you.
Thank you, sir. Any doubts?
Subject you chose, yes, this is very important for our, human health and these things. It's excellent. We have, more plants.
Yes, sir.
Namaste everyone. Today I am presenting a research named Diversity of Birds and Eudiolia. It is a comparative study. So I would like to start by uh, telling there is a shloka in Atharva Veda. It tells how important nature is for us. It is telling that may our deeds protect nature in that shloka. So our sages knew how important nature was. They maintained it until industrialization happened. They after industrialization happened, nature is not in its full form now. Um, so birds are also a part of nature, and they are playing a very crucial role in helping nature. So birds which eat fruits, they do not digest the seeds. So they'll have the seeds where and when they excrete, the seeds will fall in some area which has no plants or no trees, and a new ecosystem will start there by this one sapling. So they also help us in a way. They control pest population because it's their diet, but they also help us by controlling the pest and insect population. So there are also bioindicators, as you can see. Bioindicators are organisms which tell us the health of an ecosystem. So uh, here, the birds are also bioindicators. So my research will be my research goals are about learning the health of the ecosystem in Medieval Lake, and I will be comparing it with the paper of 2016, which was conducted by Ujula and Visa. So these are also the goals to identify the species at Edu Lake and to create a comparative report of the bird species also found in there. Both of these are my research goals. So continuing, this is the study area. This is Edu Lake, as you can see. The green part, green outline part is the tree part, and the blue outline is the water. As you can see, around it, it's a lot of urbanization happening. A lot of buildings are there, and there must be a main road next to it. So a lot of pollution is happening next to it, but still it helps 22 species of birds live. So continuing. There are a lot of plastic bags and plastic covers on the shore of the lake. So it is also polluted by littering materials and methods. I use the method, convenience sampling method, to find the bird species. 1.5 hours were spent in the lake for 2 days a month. So now 3 months have been completed. If I was going in the morning, it would be at 6:30 to 8, and in the evening 5:15 to 6:30. Yes. So my results are I have spotted 22 species, and I have classified them into 4. So the highest here is wading birds. Wading bird species are the most common ones to be seen here, and the least is birds of prey. Other common birds and aquatic birds seem to be at a steady number. Now, the discussion based on my research, my species are found, number of species found are 22. Down species are wading birds. Species spotted only in the evening. I did not see any painted storks at the morning. And painted storks were not in large numbers, only one was there. And in the last week of observation, I saw a younger one joining the big one. So, species spotted only mostly in the morning is kingfisher. I didn't see any black kite nesting or hunting in the lake. And birds least in numbers here are raptorial birds. Now these are some of this is the checklist. I have put the scientific name, common name, family name, and IUCN status. So uh, there are painted star, spot builder, pond heron, etc. So this is the checklist. Coming to my comparison, here I have spotted 22 species, whereas Rizla and Visser have spotted 35 species. I have spent 1 month or 3 months actually, and he has spent 6 months. Uh, Different species. Compared to my studies, he found 18 different species, but I found 6 different species compared to his study. Here in 2016, fewer species were found very dominant and in large numbers, but it has changed to wetland species. Wetland species are same as wading birds. So, Raptorial, he has seen 2 birds. He has spotted the black kite before, and I have seen one, which is the brown egret. So, coming to my conclusion, this lake has a very special features in Edu Lake. It has a very special feature. When Ganesh Chaturthi comes, they have a separate pool to submerge Ganesh idols, so it does not pollute the other parts of the lake. And there are many non-biodegradable plastic covers and other non-biodegradable things surrounding the lake. So it is coming to later in itself. And I have spotted fewer frugivorous birds than Rizal and Wizard. And there are many biodegraders, but decreased. So now these are the further studies that I am planning to do, finding out why the dominant species has changed. For more species, I think because there are a lot of less nut and fruit trees near the lake or surrounding the lake, because there are more buildings and other species kind of trees. And I want to write another comparison. Uh, based on all of the years from 2006 to 2024. So these are my references. Uh, these are some of the photos I have taken, and I thank all of my teachers, and I thank IISC for giving me an opportunity to communicate about this lake.
Watching over a period of watching 8 years apart, 2016 and 2024, is really a highly painstaking work that only a highly devoted can understand. highly appreciate you for this painstaking and your devotion for watching, especially in that area, Rio Lake. Your observation for changes in the visiting of the birds and also the kind of birds that have been coming to this lake. Very beneficial for the Bangalore people at large. Please keep it up. Thank you for your work.
Thank you, sir.
We'll have participation certificate distribution now. I will call upon the judges to come and hand over it to the students.
ನಿಮ್ಮ ವಯಸ್ಸಿನಲ್ಲಿ ನಾವು ಈ ಎನ್ವೈರನ್ಮೆಂಟ್ ಅನ್ನ ಪದ ಕೂಡ ಕೇಳಿರ್ತೇವೆ. ನೀವು ವಾಟರ್ ಅನಾಲಿಸಿಸ್, pH ಅನಾಲಿಸಿಸ್, ಟರ್ಬಿಡಿಟಿ, ಈ ಪದಗಳು ತಲೆ ಒಳಗೆ ಇಲ್ಲೇ ಇಲ್ಲ. ಯಾಕೆಂತಂದ್ರೆ ಪರಿಸರ ಚೆನ್ನಾಗಿ ಇಂಗ್ಲಿಷಲ್ಲಿ ಮಾತ್ರ ಸಿಸ್ಟಮ್ ಇಸ್ ರಿಮೆಂಬರ್ಡ್ ಓನ್ಲಿ ವೆನ್ ಇಟ್ ಗೋಸ್ ರಾಂಗ್. ಎನ್ವೈರನ್ಮೆಂಟ್ ಚೆನ್ನಾಗಿದ್ದರಿಂದ ನಾವು ಅದ್ರ ಬಗ್ಗೆ ತಲೆ ಕೆಡಿಸ್ಕೊಳ್ತಾ ಇದ್ದೀವಿ. ತನಕ ನಾವು ಒಂದು ಹಾರ್ಟ್ ಇದೆ ಅಂದ್ರೆ ಮಾಡ್ಕೊಳ್ತೀವಿ. ಹಾರ್ಟ್ ಯಾವಾಗ ಜ್ಞಾಪಕ ಆಗುತ್ತೆ ಅಂದ್ರೆ ಅದು ಅಡ್ಡಾದಿಡ್ಡಿ ಓಡಕ್ ಶುರುವಾಗುತ್ತಲ್ಲ, ಆವಾಗ ಒಂದು ಹಾರ್ಟು, ಇತರ ಹಾರ್ಟು, ನೊಕ್ಕರೆ ಹಾರ್ಟ್ ಒಳ್ಳೇದು ಅಲ್ವೋ, ಈ ತಿಂದ್ರೆ ಹಾರ್ಟ್ ಒಳ್ಳೇದು, ಸದಾ ಹಾರ್ಟಿಂದ ಯೋಚನೆ, ಯಾಕಂದ್ರೆ ಆ ಹಾರ್ಟ್ ಕೆಟ್ಟೈತಿ. ಹಾಗೆ ಇವತ್ತು ಯಾಕೆ ಇಷ್ಟೊಂದು ಎನ್ವೈರನ್ಮೆಂಟ್ ನಿಮ್ಮ ವಯಸ್ಸಿನಲ್ಲೇನೆ 6th, 7th, 8th ಸ್ಟ್ಯಾಂಡರ್ಡ್ ನಲ್ಲಿ ಎನ್ವೈರನ್ಮೆಂಟ್ ಬಗ್ಗೆ ಇಷ್ಟು ಕನ್ಸರ್ನ್ ಇಟ್ಕೊಂಡು ಇಷ್ಟು ಡೀಪ್ ಆಗಿ ಸ್ಟಡಿ ಮಾಡ್ತಾ ಇದ್ದೀರಿ ಅಂತಂದ್ರೆ ಎನ್ವೈರನ್ಮೆಂಟ್ ಕೆಟ್ಟಿದೆ. ವಿ ಆರ್ ದಿ ಪ್ರಾಬ್ಲಮ್ ಅಂಡ್ ವಿ ಹ್ಯಾವ್ ದಿ ಸೊಲ್ಯೂಷನ್. ಸಮಸ್ಯೆ ಮೂಲನೂ ನಾವೇ, ಅದರಿಂದ ಸಮಸ್ಯೆಯ ಪರಿಹಾರವು ನಮ್ಮಲ್ಲೇ ಇದೆ. ಆದ್ದರಿಂದ ಪ್ಲೀಸ್ ಕಂಟಿನ್ಯೂ ಯುವರ್ ಪರ್ಸಿವೇರೆನ್ಸ್. ಈ ಎಫರ್ಟ್ ಬರೀ ಅದೊಂದು ಪ್ರೈಸ್ ತಗೊಳ್ಳೋದಕ್ಕೆ ಇದಕ್ಕೆ ಅಲ್ಲ, ಇದು ನಿಮ್ಮ ಸ್ಕೂಲ್ ಎನ್ವೈರನ್ಮೆಂಟ್, ನಿಮ್ಮ ಸರೌಂಡಿಂಗ್ ಎನ್ವೈರನ್ಮೆಂಟ್ ಚೆನ್ನಾಗಿ ಆಗಬೇಕು ಅಂತಕ್ಕಂಥ ಒಂದು ಕನ್ಸರ್ನಿಂದ ಪ್ಲೀಸ್ ಕಂಟಿನ್ಯೂ ಯುವರ್ ಪರ್ಸಿವರೆನ್ಸ್, ಯುವರ್ ಸ್ಟಡಿ and your interest. Wish you all the best and wish the country a very good and clean and healthy environment through you. Thank you very much.
Ujjwala M and team. Acharya, Rupika N, Chandana J. Gopeshri S, Saraswati Yadav R, and Shashank S. Halima Manan, Harsha S, and Yutika Yamar. Nandini M, Chitra D, and Disha D. Eknath GP, Jeevan M, and Sanjay S. Sanjana K, Vivan Raj and Manoj M. Shivamakking Selvakumar. Stanley Kapurkar. RSV Sai Prasad and Jitin B. Ishka Bedi. Anita Krishna. You know the order, right? When the one name, other team please be ready, okay? Inishamba R and Lakshmita R. Eksha S and Saida N. Vaibhav Karthik R and Purnesh R. Ratna D, Aditi HG. Isha C. Near the dais so that it will be fast. Maitreya HR and Chinmay Mohan. Ananta M. Rao. Tejas Kumar Sharma. Abhinav JR. Yakta Vivek, Pratham and Arjun. Rishank, Manish and Sri Sarvesh. And Rajashri. Adya Ram and Hitaishini. Lasya and Clara. Atri. Bhargavi M and Advik GN. Ashara and Vindhya M. Swami NV and Ulas M. Samanvi H, Siddharth H and Krishna. Mokshita M. And Adya A. HM Kushal and KM Ganesh and HF E. Harshul and Tanya. U Tejasvi and KV Aditi.
Thank you, judges. Thank you, everyone. So you can all discuss now and please go to B.S. Acharya. Okay.
Guys, I request all to go to BS Acharya auditorium. Please go calmly, don't make any noise. Try to occupy the seats there, okay? Opposite to you. Please go ASAP.